Right. So I believe most of you have done anatomy and physiology, if not all of us. So it should be like a quick review. Okay. Remember, the test will consist of some biology, you have some physics, and then you have some chemistry. Most of the time when I take the poll, a lot of people haven't done physics. Okay. So I assume the sample size is very similar. Okay. So when you get to physics, I'll go slowly, go over a lot of the materials that you need to know for the exam. So don't worry at all. You will still be able to get it right when we get to physics. Okay. But most people have done chemistry and biology, so it's a lot bit easy to review that. So let's look at the first topic that usually they ask questions on, and that is the cell structure and function. Okay. I hope you can all see what I have on the screen. Everybody's okay with that? Can you all see?
Yes, I really can see.
Okay.
Yes, I can see it.
All right. If you can see, please alert me. Okay. Good. It's the same material so that you have on the handout, as I said. So it may be clearer at your end. So basically, we say that all organisms contain one or more cells which are capable of carrying out life activities needed by the organism. So the cell is a unit structure in all living things. The cell is a unit function of all living things. And so all cells come from pre-existing cells. That is what is called the cell theory. The cell theory. Okay. One exception is virus. Virus lacks the typical cell structure. So virus is not a cell. It's just a particle, not a cell. So a quick overview of what a cell is made up of. So basically, a cell is made up of a protoplasm and then the cell membrane. Okay, protoplasm and cell membrane. So if you have the cell, I'm going to draw that here. Let's see. Okay, so the cell looks like this. So that's the cell membrane. And then you have over here will be the protoplasm. Everything here is a protoplasm. So the protoplasm is made up of cytoplasm. So if you have the nucleus here, this together with that form the protoplasm, right? And then this is the cytoplasm. Okay. That's what it is. So if we take out the nucleus, they have a cytoplasm. Okay. So the cytoplasm and the protoplasm, sorry, cytoplasm and the nucleus, which is this, form the protoplasm. Okay. That's what we have here. Okay. Now, what you should know for the test basically is the structure and function. You have to know this very well, the structure and function, structure and function. That's the way you should study for the test. And I have a typical structure of the cell here. So we'll go through that first. Yeah. So when you talk about the nucleus, which is the nucleus, one thing I have to remember of the nucleus is the fact that it contains the DNA. So the DNA in the nucleus, very important to remember that. My pen is acting up a little bit, so I'm moving that fast. Give me a second. Let me look at my pen again. Sorry, my pen is slowing me down. So have a little patience on that. Okay, I got it. All right, so the nucleus, once you mention the nucleus, remember DNA. Just a second again. Okay, let's go on. I'll try and fix this as we go. I wanted to write a little bit. Right, the nucleus, once you talk about nucleus, let's go back here. So the nucleus is a control center for the cell. That is a nucleus. And then once you mention the nucleus, remember that it contains the DNA, which directs the synthesis of proteins by the cell, right? So that's a function of the nucleus. And then the mitochondrion is a powerhouse of the cell. It carries on the process of cell respiration by converting glucose to ATP energy. Okay. And then you have the ribosomes, which are found on the endoplasmic reticulum and free within the cell. So there are two types of ribosomes. One of them is the smooth ER, and then the other one is the rough ER. So you have smooth and rough ER. Okay, I'll come back to that again. Okay. And then the function of the smooth ER is for lipid synthesis, right? A smooth one for lipid synthesis. And then the rough ER is for protein synthesis. That's for protein synthesis. So there are two types. Okay. And then you have the function of the endoplasmic reticulum is to transport proteins within the cell. So it's like a transport channel within the cell. And then you may also have food vacuole within the cell that store and digest food. Okay. Also within the cell, you have the lysosomes. The lysosomes, they contain digestive enzymes that break down substances in the cell, okay, for the lysosomes. And some cells will have what we call the contractile vacuole. Not every cell will have this, all right? But some cells have that, especially a cell like the amoeba. Amoeba is a one-cell organism, right? Has what we call contractile vacuole. And the contractile vacuole, it pumps out waste and excess water from the cell. Okay. And then also, we mentioned the cell membrane already. The cell membrane is selectively permeable. So it regulates the movement of materials in and out of the cell. It controls what enters the cell, right? So these are some of the structures, or what's called organelles, that you have to know within the cell, especially for this particular test. They like to ask a lot of questions on the cell. Okay. Yeah. So let me scroll down to the cell again for us to look at that. So these are the structures I'm talking about. This is a mitochondrium, like a sausage structure, mitochondrium, right? It's a powerhouse. So any question that you see ATP, you have to think about the mitochondrium, ATP, mitochondrium, right? So let me see if my pen is back again. I can write. Okay. All right, good. It's working now. So I mentioned this DNA. Think about that when you see a question DNA. Anything that has ATP in a question, think of the mitochondrium, but as a powerhouse of the cell, energy production, right? Then when it comes to the cell membrane, so cell membrane, the word that is selectively permeable, selectively permeable membrane. Okay. In other words, it only allows certain things to go in and out of the cell. Okay. So if you take the cell membrane, I believe in your class, anatomy physiology class, you were told that this is a bilipid layer. Bilipid layer, right? So made up of 2 layers of lipid. So if you remember from your class, I believe you all remember it's shaped, it drew this diagram like this for you. You have the head and the tail. So this is a phospholipid. So that's the phospholipid, which has a head and tail. Phospholipid. So it's made up of a phosphate and a lipid portion. Phosphate and lipid portion, right? The head portion lasts water. It is a head, and the tail portion heats water. So this is said to be what hydrophobic, that's the tail part, and the hydrophilic, that's the head part. Okay. So since there's water outside the cell and inside the cell, the lipid will be hiding itself in the middle. So we have another layer where the lipid is hiding in the middle because it hates water. So they hide. So this is how the cell membrane looks like. It's a 2-layer of phospholipid. The lipid is inside, hiding here, lipid part here, and the phosphate is outside. Okay, right. So for something to go in and out of the cell, there are some channels through which the materials go through. And those are protein channels. So you have a channel like this that goes through. So you have a protein that goes through the whole membrane, which acts like a channel protein. Okay, so water can go through and other substances can go through. So it selects what goes through the cell membrane. That's why it's selectively permeable. We'll talk about transport along the cell shortly. Okay. And then the endoplasmic reticulum, which I mentioned, there's the ER right here. The ER. So the one that has ribosomes on the surface, we call them the rough ER. Endoplasmic reticulum. And Fori said that this was protein transport, as we said. So protein transport. Okay, that is the function of the rough ER. Okay. And that one has the ribosomes on the surface. We have the ribosomes on the surface. So the ribosomes will be here. The ribosomes will be somewhere here on the surface like this, ribosomes. Okay. So that's why it is rough. And in the ribosomes, we have to remember the function of the ribosomes is to make proteins. Okay. Ribosomes make proteins. So ribosomes. So the function is protein synthesis. So if you see this, think about protein synthesis. Okay. Protein synthesis. So that's the function of the ribosome. So structure and function, that's how we should approach this type of problem. Okay. So the one that doesn't have ribosome on the surface, like I'll draw that here, so you have no ribosomes on the surface, and that's what we're going to call this smooth. Endoplasmic reticulum. Okay, that's a smooth one. And the smooth endoplasmic reticulum, the function is basically for lipid synthesis. That's the function of the smooth endoplasmic reticulum. It makes lipid. It makes lipid, right? So on the test, you could have a question like, somebody was looking under the microscope, and the person noticed that the cell has a lot of smooth endoplasmic reticulum. What is the likely function of this cell? Right, then you have the options. So you have to look out for something that is involved in lipid synthesis, right? So something like the liver has a lot of, the liver cell has a lot of smooth endoplasmic reticulum in it. So it's involved in lipid synthesis, right? So think about that like that, okay, right? And then, so basically, so this more of like animal cell, right? There's difference between the animal cell and the plant cell. So note some of the differences. So if you go to number 9, which I have here, number 9 here, and number 10, I made mention of differences, right? And one of the difference is that the cell, animal cell has no chloroplast. The plant cell has chloroplast. So I put here, found only in plant cells and algae, the chloroplast. And remember, the chloroplast contains chlorophyll. The chlorophyll is a green coloring substance in the plant, okay? And that carries on the process of photosynthesis. So you see chloroplast in the question, think only of a plant cell. So the question says that somebody's looking at the microscope and the person notices that the cell has a lot of chloroplast. Which of the following is the person looking at? Let's say cell from the cat, cell from the mouse, cell from an onion, then the last one maybe cell from a dog. You have to go for the onion because that is the plant. So chloroplast in plants only. Again, if you see a question that has cell wall in it, cell wall, think about plants. So you look for the answer that is a plant because that is only found in plants, only found in plants, okay? And another word that you may come across the cell plate is somebody observes a cell plate under the microscope, then the person is looking at a plant cell because the cell plate becomes the cell wall. When the cell is dividing, it becomes a cell wall. So these are some of the exceptions that you should look out for, especially on the test, the difference between the plant cell and then the animal cell. Okay. Right. Does anybody have any question on this first part before we move on. We'll look at some questions in the book as we go on. Yes, you have a question?
No.
Oh, you're fine. Okay. All right. Anybody has any question on the cell structure and function? Okay. All right. Feel free if you have a question, please stop us and then we'll discuss that. All right. So the next one I want to look at is movement of substances across the cell membrane. Movement of substance across the cell membrane. Okay. And there are 5 main ones that I want to discuss. The first one is diffusion. Okay. So what is diffusion? Now, diffusion, or what we call the passive transport, is a movement of materials from a region of higher to a region of lower concentration. So the substance moves from higher to a lower concentration, right? So usually I like to draw a diagram that looks like this. It looks like, let's put it here. Okay, let's see. For some reason, I don't know why today my pen is so slow. Okay, let's see. Okay. So you have, let's say, particles here. So this is crowded particles, and then less crowded on this side. Okay. So diffusion basically is a movement of a molecule from a region of high concentration to a region of low concentration. So the molecules move from high to low, right? So I use a word like a crowded room. People want to be free, so they start moving outside where there's less people. That is simple diffusion, okay, or passive transport. No energy is needed over here. Okay, so that's a key. No energy needed. Passive, no energy needed. Okay, right. And an example would be when a bottle of perfume is opened, the scent spreads throughout the room. That is an example of passive transport or simple diffusion. Simple diffusion. Okay. Then another example is if you have ink, let's say water, and you put a little ink, or let's say blue ink or red, or a dye. Okay. After some time, what happens? The whole water becomes blue because the molecules are concentrated here. They diffuse out to a less crowded area around it. So the whole thing now becomes blue after some time. That is simple diffusion. Okay. So this is one of the questions that they like asking a lot. It's one of sometimes they give a scenario, like they give an example, and then they ask you which type of movement it is. Okay. So that's 2 examples here. Okay. Another example that I can give is breathing in and out, right? So breathing in, we have more oxygen outside than inside the lungs. So oxygen diffuses into the lungs easily by simple diffusion. And then inside the lungs, we have more carbon dioxide than outside. So because you have carbon dioxide crowded within the lung, it diffuses out, comes out to the less crowded area. Okay, so that, which is the environment. So that's another example of simple diffusion. So they can give you any of these scenarios and they ask what it is. Okay, good. Then the second one is the active transport. Active transport. So active transport is like the reverse of diffusion, right? So it is movement of molecules from a region of lower concentration to a region of a higher concentration. So those are the keywords, from a lower to a higher concentration. So take note of those keywords. Region of lower to a higher. And this time energy is needed, and the energy comes in the form of your ATP. Okay. Okay. So that is active diffusion. Energy is needed. So the diagram form, it will look like this. The molecules, few molecules here. You have a lot of molecules on the other side. Now this will diffuse from low to a higher concentration. So the scenario that I always use is like a crowded room. So this is a crowded room. You have people outside. They still want to come in. How can we put them in? You have to maybe push them. And pushing them, you need energy. Energy is needed here. And that is the form of ATP. Okay. So that is the active diffusion. Energy is needed. And a typical example that I have here is what we call a sodium potassium pump, the ATP pump. I'm sure you've heard of the sodium potassium ATP pump. The pump sodium Na plus that sodium potassium ATP pump. Okay. It's an example of active diffusion. All right. So sodium is pumped out of the cell by active diffusion, and the potassium is pumped back into the cell by active diffusion. After an impulse travels along, let's say, the axon, the neuron, right? The sodium has to pump back outside, and the sodium potassium has to pump back in. And this needs energy to do that. And that's a form of active transport. Okay. So remember, active transport, energy is needed. Okay. Then the third one is osmosis. Osmosis, that's another favorite question that they like to ask. So osmosis is the flow of water molecules from a less concentrated solution, which we call the hypotonic solution, to a higher concentrated solution. And that solution we call the hypertonic solution through a semi-permeable membrane. So the key word that you have to remember, you need to have a semi-permeable membrane somewhere. Semi-permeable membrane. If there's no semi-permeable membrane, you cannot have osmosis, okay? Because that's the selection. It allows only water to pass through the semi-permeable membrane that we're talking about here, okay? So the water flows from less concentrated solution to a more concentrated solution through a semi-permeable membrane. And an example is when you have the red blood cell and you place it in water, the water flows across the cell membrane into the cell because you have dissolved salt in the cytoplasm. Okay. And another example is water flows from the soil into the roots of plants by osmosis. By osmosis. Okay. So that's what happens in the roots. Okay. So you can always do these experiments if you have kids who want to do a science experiment. One of the easiest experiments they can do is the osmosis experiment. And this is how it will look like. So if you take potatoes and then you scoop the inside and turn it into like a cup. So let's call this potato cup. And then let's say water in a bowl. Now we can put in salt solution here. Salt solution. And then leave this for some time. If you come back, you realize that the level of the water will increase. It's going to increase. It's going to go up. Why? Because the cell, the potato cup, the potato tissue here is acting as a semi-permeable membrane. So this is the semi-permeable membrane. So it allows only the water to go in. So water goes in. And then the level rises. So this is an example of osmosis working. So this is osmosis working here. So the water here is the hypotonic solution, less concentrated, and then the salt solution here is the hypertonic solution that we're talking about. So osmosis is the flow of water molecules from a less concentrated solution to a more concentrated solution through the semipermeable membrane. If this is not there, if you don't see this in the question, then you cannot say it's osmosis. Okay. So look out for semipermeable membrane in the question. Okay. So take the root of plants, as I said, if you take the root of plants, now the membrane will act as semipermeable membrane. It is the root, and then you have dissolved salts here. So the water goes in by osmosis, okay, because it's less concentrated outside than inside of osmosis. The same with the red blood cell. You take a red blood cell, you put it in water, as we just discussed here. It contains a lot of dissolved salts. So the water flows into it, so this red blood cell, by osmosis. Okay. So you can have a question where they tell you that red blood cell was put in water. After some time, the whole water turns red. What's happening? It's a form of osmosis. The water went into the cell, and then because the cell cannot, the cell will expand. But when the cell cannot expand forever, it will end up rupturing. Okay, to rupture, and then the whole solution will become red. So that is a form of osmosis occurring in there. It's a form of osmosis. Okay. And then the next one is called the facilitated diffusion. So in facilitated diffusion, you're talking about a certain substance can move into the cell by simple or passive diffusion. It goes inside easily, into a cell easily. As it goes inside the cell, it creates opportunity for other substances to follow it freely into the cell, right? So it goes in easily, and then it pulls or draws other substances into the cell. A typical example would be the sodium, the movement of, so we come here is a movement of glucose into the cell. So glucose, like sodium, so needs sodium to go into the cell, right? Because sodium can go to the cell easily. So as sodium goes into the cell, glucose follows it. Okay, so sodium goes in, and then the glucose follows it. So the glucose, the sodium has facilitated the movement of glucose into the cell. Now, this is very important, especially since you're going to become nurses. If somebody comes in and the person has diabetes, a person has what we call the diabetic coma. You know, sugar level is so high, and then the person is getting has passed out. You don't just go and give the person insulin, even though the sugar level is high. You don't start with that. The person has to be hydrated first. You have to give the person normal saline. The normal saline, because you want to increase the level of sodium in the person blood. So you give normal saline first, and when the sodium level has been corrected to the normal level, then you can give the insulin, because the sodium is needed for the glucose to go into the cell to be metabolized. Okay, so that is an example of facilitated diffusion. Okay, facilitated diffusion. Okay. All right, so glucose follows sodium as it enters the cell by passive diffusion. Okay, and then the last one I want to talk about is endocytosis. Endocytosis. Endocytosis, this is the process by which the cell engulfs or captures substances into the cytoplasm. Okay, endocytosis. There are two types of endocytosis. The first one here is the pinocytosis, and then the second one is called phagocytosis. cytosis. Panocytosis, literal meaning the cell is drinking water. So the cell takes in the liquid substance. So you can say drinking, cell drinking. And here, literally, we can say that the cell is eating, like the cell eating something. So it picks up a solid substance. That's the way we can remember them: cell drinking and the cell eating. Okay. So let's take something like amoeba, the one cell organism that looks with a nucleus. It sees a food particle, and then it will send part of its cytoplasm around the food particle like this. And then this will fuse and then to capture the food substance. And eventually this will become what we call a food vacuole. Okay. So this is endocytosis. It captures the substance and takes into the cell. If this is a liquid, then it's pinocytosis. If it's a solid, then it becomes phagocytosis. Okay. So this is how the food vacuole is formed. So this is a vacuole. This is how it's formed. Okay. Or if it's a bacteria, let's say the neutrophils, the white blood cell. If it's a white blood cell and it's a bacteria, this is how it captures it. It's a phagocytose the bacteria. And then lysosomes can bind to this, and then they release the substances to they release the enzymes into it and digest it and destroy the bacteria. Okay. So this is endocytosis. Two types: pinocytosis and phagocytosis. Okay. Right. So those are two things I should note for this. Okay. So for the movement of substance across the cell membrane, try and know that very well. Okay, in the cell structure and function. They always ask questions on that, always. Okay. Does anybody have any question on the movements?
No.
Okay, so far so good. Okay. As I said, we'll see the application in the textbook soon. All right, so I have that here, so that's fine. And then exocytosis is moving the substance out of the cell, getting rid of the substance. Okay. So it's backwards, right? Pushing it out. Okay, good. All right. The next thing I want us to discuss is the organization of the organism. Okay, organization. Okay. We all know that we have the cell, and they have a group of cells that work together or they perform similar function. It becomes a tissue. Similar tissues working together form an organ. Organ form the these different organs working together form the system, and then they have the whole organism, right? It's a system organism. Okay. So that's the level of organization that we have here. Okay. I want us to look at the different types of tissues that you have. So the types of tissues. We have epithelium, or the epithelial tissue. That's the first tissue. And they have connective tissue. And they have the nervous tissue. And then the muscular tissue, or the muscle tissue, the muscles. Okay. So those are four tissues that we want to look at. So let's begin with the epithelium. So the epithelium over here, so say tissues that line or cover body surfaces. We call them epithelium or epithelial tissues. Okay. And you can classify them based on the layers. If you have one layer, we call that simple epithelium. If you have multiple layers, we call that stratified epithelium. And there's one that has false layers, and we call that the pseudo epithelium. Okay, some people call it pseudo stratified epithelium or the pseudo epithelium. All right. So let me see if I can draw what I'm talking about here. So every So simple epithelium, it can be one cell. So one cell, that means that you have what we call the basement membrane, and then you have the cell on it like this, with the nucleus in there. Okay. So this is called a basement membrane. Okay. And the cell on it. So one layer, that is simple epithelium. Stratified, you have a basement membrane, but then you have more than one layer. Another one layer on top of this. So that becomes a stratified epithelium. Okay. And then what the pseudo-stratified is a false layer. It appears as if you have more than one layer, but it's actually a single layer. So you have a basement membrane, and all the cells arise from the basement membrane, but they overlap. So you have one like this, and I may have another one that appears to be another layer, but the same layer, because they all come from the same level. Okay. So this is a false layer. False layer. Okay. So that's the pseudo-stratified epithelium. Okay. All right. So that's one classification of the epithelium based on the layers. Usually in areas that we want protection, that's where you have multiple layers. For example, the skin. If you take the skin, you need a lot of protection. So to have, it would be like a stratified type of epithelium, okay, many layers, okay, where you have wear and tear, many layers. If you want quick diffusion of substances, like in the lungs, the alveolar, the air sac, the lining, is just one cell, one layer of cells. So it's a simple epithelium because you want the oxygen and carbon dioxide to diffuse quickly. So it just be one layer of cells. Okay, right. So that's how you can remember them. Protection, multiple layers, where you want quick transport, like diffusion of gases, simple epithelium, single layer. Okay, right. And then you can also classify them based on the shape. And the shape, the first one that I have here is called the squamous epithelium. So squamous epithelium, these look like flat cells. The cells are flat, like a pavement. So you have the basement membrane, and then you have the cells like they have flat cells. Okay. They have a squamous type of epithelium, squamous. Okay. Then they have those that are like cubes. You call a cuboidal epithelium, and then the nucleus will be round. They look like cubes, so cuboidal epithelium. And then the last type is known as the columnar. They look like pillars, like tall cells, like columns, tall columns. And then the nucleus will be like, the long axis will be perpendicular to the surface. So like pillars, columns. Okay. So these are the diagrams that you should look at. Sometimes they draw the diagram for you and it asks you to identify, which epithelial tissue it is. So that's why I do a lot of drawings, so you can see them. Now, one thing I have to remember about these tissues is that some of them have hair-like structures on the surface. Like hair-like structures. They like asking this question. Now, those structures are known as the cilia. So if you have the cilia on the surface, then we call it is a ciliated epithelium. Ciliated epithelium. Now, where do you find ciliated epithelium? It's good to know where you find some of these cells. For example, the ciliated epithelium, we can find one in the respiratory system. respiratory tract. You can find it. One of the areas is here, the columnar epithelium, the respiratory tract. The cilia beats to move substance on the surface, right? So let's say you have mucus in the respiratory system, the cilia will beat and move the mucus towards the throat so that you can cough it out, right? So it's very important in the respiratory tract. Another place where you can find another ciliated epithelium, usually the columnar one. The columnar can also have cilia on the surface. You can find that in the fallopian tube. You can also find, but that one is a columnar type of ciliated epithelium. So that moves the egg towards the uterus, you know. So so this is in the fallopian tube. So those are two things if you can remember. Ciliated epithelium, they like asking about the ciliated epithelium too. So you can put that down somewhere to remember. Respiratory tract, very common question. Okay. Great. Then the connective tissue. For the connective tissue, that one we'll go into details along the line. We'll talk about the blood later. But for this connective tissue, no examples. Okay, sometimes they ask about example, which are the following is a connective tissue. Then they give the list. So you should identify them. So remember, bone is a connective tissue. Blood is a connective tissue. Adipose tissue, which is fat tissue, is a connective tissue. Cartilage, connective tissue. They have the connective tissue proper. When I say connective tissue proper, you're talking about, let's say if you take the skin, under the skin, you have the connective tissue proper contains other things. It holds the other structures together, binds the structures under the skin. Those are connective tissue proper. And they have tendons and then ligaments. Tendons, they connect they are the end of muscles, right? And then ligaments, they hold bones together. So these are examples of connective tissue that you should be able to remember. When you see the question, you should be able to identify them. Okay. Great, so we'll leave that for now. There's examples that you should know. And then you have the nervous tissue. The third one is the nervous tissue. So you should know this structure also very well. So I number them 1, 2, 3, 4, so you can identify them. If you give a diagram, you will identify each section. So the basic unit of the nervous tissue is the nerve cell or the neuron, right? The neuron or the nerve cell. So we can call this the neuron. Or the nerve cell. And then the nerve cell has nucleus. So that is one. This is a nucleus. Right. This will be what we call the dendrites. And then 2 is called the cell body. Or some people call it the cytin. You say cytin, the same thing. Then the long structure here is the axon. The axon. The axon is like the longest dendrite. So it carries the nerve message or the impulse to the end. You know, so the impulse generated here at the end dendrites, then travels along the cell body, gets to the axon to the other end here. So at the end, number 4, this is the terminal dendrites. terminal dendrites. Okay. Or some people call the end brushes, as you have the end brushes. Okay. So this way you have the neurotransmitter released at the end here, neurotransmitter is released. So that will stimulate another neuron. And then around the axon, you have what we call the myelin sheath. You have the myelin sheath. Okay. Now the function of the myelin sheath is to insulate the axon. It protects the axon. Okay. So that it can easily transmit the electrical stimulus. So that's over here, the myelin sheath, fatty sheath that surrounds the axon. It provides electrical insulation for the nerve and also allows for rapid transmission of nerve impulse. Okay. So what this means is that if you have a neuron that has myelin, the transmission of impulse is faster. Because this is what happens. The impulse jumps from, let me mark something here. There's something called the node over here. You got the node. Node of Ranvier, somebody's name. Node of Ranvier. Okay. So there are breaks between the myelin sheath called the node of Ranvier. So these nodes, if they are present, then the impulse is fast because the impulse, it jumps from one node to the other. The transmission impulse jumps. So it's like galloping, so it's faster. If there's no myelin sheath, then the impulse has to travel along the whole length of the axon, which is longer. Okay. But this one jumps from node to node, so it's like galloping. So it's fast transmission. Okay. And also remember that the neurons are not joined together. There are gaps between the neurons. Okay. And the gap between the neuron is what we call the synaptic gap or synapse. There's a gap over here between neurons. So this is another key thing that you have to remember. Synapse is a gap between neurons. Gap between neurons. Okay. And then one more thing that I'm going to add here, the myelin sheath, I'm going to draw something around it like this and put a nucleus on it. This is known as a Schwann cell. Schwann cell. The Schwann cell produces myelin sheath, right? It produces myelin sheath. So Schwann cell, I'm going to write that here. The Schwann cell, this produces the myelin. So that's its function. Okay. So it's around the myelin over here. Okay. All right. So this is the basic structure of the neuron that you should know. So if they give a diagram and they number them, you should identify what each part does. Okay. And then the last type of tissue we want to look at is a muscle tissue, the muscle tissue. As I said, please look at the diagrams carefully. The diagrams are very important. They give a lot of diagrams that you should be able to identify. Okay. So we'll do the same thing for the muscle tissue. So for the muscle tissue, there are three types: skeletal muscle, cardiac, and a smooth muscle. Three types. Okay. So let's look at the skeletal muscle. Now the skeletal muscle are attached to and they move bones. Okay, so attached to and moves the bones. And the diagram of skeletal muscle, when they draw that for you, should identify them too. You see, it looks like this. It looks as if you have some parallel structures, then you have fine lines in between, fine lines running through those parallel structures. Under the microscope, if you get a chance, you can look at that on what do you call it, on the internet, the diagram, beautiful diagram. If you look at the microscope, it looks like this. These are called striations. And they have the nucleus right there. Nucleus will be there. Nucleus. Nice pattern. So these are called the striations. So that's why another name for the skeletal muscle is a striated muscle. Striated muscle. So that's another name. Striated muscle. Okay. We call the striations. Then the cardiac muscle, which you find in the heart. Cardiac muscle. The diagram looks like this. You have something that looks almost like the skeletal muscle, but then they show branching. They are interconnected. So one cell will be connected to the other cell like this. And you have the nucleus, then you have another cell. They show interconnections. So see a diagram that looks like this, then you talk about cardiac muscle. The interconnections. Okay. So found the heart. And the last one here is the smooth muscle. The smooth muscle, we find it like structures in the body cavity, like the stomach, the intestines, the uterus, and so on. Right? Smooth muscles. And the smooth muscles, they are like spindles. They are spindle shaped. So spindle shaped. So spindles, that looks like this. And you have the nucleus. So if you see these structures, then those are the smooth muscles found in internal organs. Okay. So these are the three types of muscles that you should be able to identify in questions. See this diagram: striations, striated muscle, cardiac, and then spindle-shaped smooth muscles. Okay. They like drawing some of these diagrams. Okay. So you can draw that in your notes as well. Okay. Right. So I'll pause here for 30 seconds to ask if you have any questions on these ones. So we'll cover the lesson a little bit before we look at how they're related in the book. So if I say everybody okay with the first part, few parts are we done? Are we all okay so far?
Yes.
Yes.
Good. All right. Okay. Right. Good. So let's now look at the chemistry of the cell. So the food substances. There are three food substances that I want to talk about: carbohydrates, proteins, and fat. Okay. So we all know that carbohydrates, we have what we call the simple sugars, also known as the monosaccharides, right? The monosaccharide, one sugar molecule. Know all this inside out. You got to know them very well. Glucose, fructose, galactose. Those are the simple sugars, right? So one way is to use mnemonics to help you. So or just abbreviation. So is it GFG or GGF? Something to help GGF or GFG. Those are the simple sugars: fructose, sucrose, and galactose, the common ones. And out of this, we can form the disaccharides. Disaccharides are two sugar molecules. And they are the sucrose, maltose, and lactose. Those are the disaccharides, one or two of these. Okay. And you should know what each of them is composed of, because when we talk about digestion, we're going to work backwards. If somebody eats lactose, what will be the end product of digestion? If somebody eats glucose, a food containing glucose, sucrose, what will be the end product of digestion, and so on. So working backwards. Okay. Lactose, so that can find in milk. So if somebody drinks milk, what will be the breakdown? If somebody sucrose in cane sugar, sugar cane, right? That's sucrose. If somebody eats that, what will they break down? Yeah. Now, how do I remember this? So I'll show you how I remember this. Basically, if I take, let me start with lactose. If I take lactose, in my mind, it rhymes with galactose. So I know it's galactose is one of them, breakdown is galactose. Then plus the other one, they all contain glucose. So I don't worry my brain about that. So I know glucose is one of them. All of them contain glucose. So lactose is galactose and glucose. When I take sucrose, for some reason it's funny, but I don't know why it sounds like fructose in my mind. Sucrose fructose. So I know it's galactose, glucose is one of them already, so I don't worry about myself. Sucrose fructose. So this is fructose. And then the last one, which is a maltose, found in like malt, it's made up of glucose and glucose. That's one, two glucose molecules. Okay. So once I eliminate lactose, and I know the other one is fructose, so sucrose fructose, lactose, galactose. Yeah. And the second part is just glucose added on. Okay. So somebody is this, that will be the breakdown of that. Somebody is sucrose to break down into this and that, maltose, glucose, and glucose. Okay. We'll talk about that again on digestion. Okay. And then you have the complex sugars. The complex sugars. And the complex sugars, remember that they're also known as the polysaccharides. This is the, another name is polysaccharides. Polysaccharides. So the polysaccharides are the complex sugars. So example is starch, cellulose, glycogen. So they are made up of sugar, glucose molecules that are joined together. So if you join so many glucose together, you have a complex sugar. Okay. We call it the polymer of glucose, polymer. Okay. So some few hints that I want you to take note of on questions. Difference between plants and animals. Glycogen is a storage form of carbohydrates only in animals. So if you see a question that's actually about glycogen, you should identify an animal in that question. It goes to animal, only in animal. Now, cellulose is only found in plant cell, in the cell wall of the plant. That's where you find cellulose. So if a question has cellulose in it, think only about a plant. Okay, so these are two things that you should not, exceptions or to the rule that you should always look out for. Okay, glycogen, only animals, cellulose, only plants. Okay. Yeah. And then I put an example of a question that they ask here. It says like about the cream cracker. Okay, so it says the question is, goes this way: somebody chews cream cracker slowly. After some time, it begins to taste sweet in the mouth. Then they ask you, what happened? You know. So I have here say the cream cracker, that's starch. It tastes sweet in the mouth because it is broken down into glucose by an enzyme called amylase. There's amylase in the mouth. When we talk about digestion, we'll refer to that again. That breaks down the glucose to, it breaks down the starch to the glucose molecules. Because remember, glucose, I said starch is made up of glucose joined together. It's a complex sugar. Okay. And because glucose tastes sweet, that's why you have it tastes sweet in the mouth. Okay. That would be the reason why it's sweet. So from the cracker will taste sweet. The question says it will taste bland, then to say bitter, is to taste sweet, and then there's one other option, like 4 options for that. So you have to choose the sweet portion. Okay, all right. So that is one example of the way they ask you the questions. The other thing that you have to know before I come to the proteins is how to test for some of the food substances. Okay, so how to test for the sugars. Right? So the table shows you, I put it together in the table form. Starch. How do you test for starch? To test for starch, you do what we call the iodine test. Iodine test. Okay, so if you add starch to iodine, it becomes blue-black. The color turns blue-black. So that'll be the test for starch. Another name for this iodine test, in questions, they sometimes use the word Lugol solution. So you see the word Lugol solution, they talk about iodine. It's iodine in alcohol. Iodine in alcohol. So plus alcohol. That's what we call the Lugol solution. It used to be used to dress wounds in the hospitals many years ago. You know, the Lugol solution. So if you add it to starch, it's turn blue-black. Then simple sugar. Simple sugar, to test for that, we can use what we call the Benedict test, or you can use the Valence test. You can work with the Valence reagents. You can use that as well, Benedict or the Valence reagents, to test for simple sugars. And if you boil it with the simple sugar, the color becomes brick red. It's like a purple substance. It has a purple color, but it changes to brick red color. Once that happens, then you know you're dealing with a simple sugar like glucose. Yeah. So know this test for carbohydrates. Okay. All right. Now we can talk about the protein. Now proteins, I want you to know that they are made up of amino acids for now. Right? Those are, just keep it simple. Proteins are made up of amino acids. So if you have an amino acid and you join them together, you have proteins. Now I'm going to mention the bond that exists between the amino acids. So let's say I have amino acid. So let's say proteins. For the chemistry of it, it looks like this. The amino acid. You have carbon, and then you have hydrogen somewhere. You have COOH. For those who have done some organic chemistry, you may have come across this. If not, don't worry too much. You just look at what I'm drawing. And then you have what we call the R group. Okay. Every amino acid looks like this. You have a carbon, nitrogen, and oxygen in it. Okay. This end here is called the carboxylic end. Carboxylic end. And then this end here is called the amino end. So every amino acid has amino end and a carboxylic end like this. The only difference is what we call the R group. The R group is made of carbon chains. In organic chemistry, this is made up of carbon, like we call a carbon chain. And this is what changes in amino acid. So it changes in amino acid. So this one varies. The R group, that's what changes. Okay. So if you have one amino acid joined to another amino acid, another amino acid, then you have a protein. Okay. And the bond between the two amino acids is what we call the peptide bond. The peptide bond. Okay. So what happens is that the amino end of another one will combine with the carboxylic end over here. It joins over here. and what is given off, and it forms what we call a peptide bond in between. Okay, we're not going to go into the chemistry of that, just a quick mention of it. So just remember the peptide bond exists between amino acids, formed between amino end and the carboxylic end. Okay, so that's the proteins. We'll talk about protein synthesis another time or later. Then the next one is lipid, fat, and oil. So remember, the lipids are made up of what we call the fatty acids and glycerol. Fatty acid and glycerol. So lipid, you have the fatty acids and glycerol. So the fatty acid in organic chemistry looks like this. You have what we call the R group, the carbon chain, and they have COOH, carboxylic end. And then the glycerol will look like this. It has 3 carbons, and they have OH here, OH. Then you have hydrogens around the carbons like this. So this is a glycerol molecule. 3 carbons and then OH attached to it and carbons around it, glycerol. So when fatty acid combines with glycerol, then you form the lipid. So lipid, fatty acids plus glycerol. Fatty acids and glycerol. So that's how you form a lipid. So if somebody eats lipid and breaks it down, digestion, it will break it down into fatty acids and glycerol. Okay. Backwards. So what happens, again, a quick one. The glycerol, the R group here replaces the hydrogen. So this R group here will replace the hydrogen here. If one of this is replaced, then we call it a monoglyceride. If you have another R group replacing this, they have the diglyceride. If another R group replaces the hydrogen here, then you have the triglyceride. You know, that's what happens. 1 mono, 2 di, and 3 triglycerides. Okay. But for now, just know that lipid is made up of fatty acids and glycerol. Okay. And just a quick test. You can for the lipid, a quick one would be lipid oil will always stain paper, right? Even when it dries, it stains it. So that's like a quick test whether it's water or oil. You know, water will dry up, oil will stain it. Okay. Right. So those are the food substances that we want to talk about for now. You know, we can add one more to this before we look at the structure of the atom. One more of this. Maybe we can look at the difference between, I wanted to talk about that another day, but we can mention that now. DNA and RNA under this topic. The nucleotides. We can mention that now. And we talk about the DNA and the RNA. Okay. So as we do this, I'm going to write down the differences. Now, all of them contain a 5-carbon sugar. And for DNA, it's called the deoxyribose sugar. And then for RNA, it's called the ribose sugar. That's where the names come from. Ribose sugar, R, and D deoxyribose. So that's the sugar molecule in each of them. Then they all have what we call the nitrogen bases. Nitrogen bases. Now the nitrogen bases, this also has the same thing. Nitrogen bases. So what are the nitrogen bases? The nitrogen bases are, one of them is guanine. So G for guanine. Then you have cytosine, C. Then you have adenine, A. And then you have what we call thymine. T. In the DNA. When it comes to RNA, you have the same thing, guanine here. You have cytosine again here. You have adenine. But then you have uracil, U. That replaces the thymine, right? So you replace the thymine here. You don't have that in the RNA. Okay. So that's the difference. So the difference, thymine here and uracil in the RNA. Okay. Now, always remember that the cytosine always binds with guanine. So cytosine and guanine always bond, and the adenine bonds with thymine in the DNA. Same thing here, guanine always goes with cytosine, and then the adenine will go with uracil. Okay. You don't have T here, so it goes with that. Okay. Right. So those are some of the things I should remember here. And one more thing I'm going to add, and that this is the fact that DNA is double-stranded. Okay, double-stranded, and this is a single strand. Okay. So if you remember the double helix, like a helical, while this one gets a single. Okay. So those are some of the differences I want you to note under these two groups. Okay. And then the last thing that they all have is the phosphates. They all have phosphates in them. Phosphates. Okay. So when you say a nucleotide, then we are talking about the phosphate plus the sugar, which is the ribose or deoxyribose, and then plus the nitrogen base. That makes up the nucleotide. These three make up the nucleotide. Phosphate, sugar, and the nitrogen base will become the nucleotide. Okay. And when you say nucleoside, there are two things here. We get nucleoside, side. The nucleoside is just the phosphate and the sugar. plus the ribose or deoxyribose sugar for the nucleoside. Okay. All right. Great. So you could have a question that goes like this. Now they give you the DNA sequence. Let's say the DNA, they give you a sequence like this. Let me make up something here. G, T, A, C. Then let's say I have another C, T, and A. Now it could be asked to transcribe this to RNA. Transcribe this to RNA. So transcription. What would be the sequence that we expect here? Would somebody like to try that for us? What would be the sequence that we expect? Anybody wants to try can go ahead. Anybody would like to try? What would be the sequence?
Would it be C, A, U?
Okay, let me write it down as you say it. So gene becomes a C, correct? Because U, correct? U. Because there's no A in the RNA. Good. And then you have A will go with
U.
U. No, hold on. Let's come back here. For T, this one.
I put A for
A,
T.
yeah, it should be A.
So
A here.
C, A, U.
U.
G.
G.
G again.
G again.
And then A and U.
A and U, correct. So that will be the transcribed form of the RNA. So they can give you the sequence and ask you to transcribe here to an RNA. Okay. So the main thing you have to remember, as you said, because you don't A, you're supposed to go with T if it's DNA, but now you have RNA, so you're supposed to go with U. You have to replace U. You should be T in the RNA. Right. All right. Great. Thank you very much for that. So that's transcription. Transcription. Right. Any questions so far on the food substances? Right. If there are no questions on that, then we can look at the concept of the structure of an atom. The structure of an atom. Okay. So you can please track the screen for that one. So said an atom is the basic unit of an element, right? And then every atom has what we call atomic number. And atomic number, we have to remember that atomic number is the same as the number of protons. Atomic number is the same as the number of protons. Now the protons and neutrons are in the nucleus. And the protons have a positive charge. The electrons have a negative charge. And then they are located in orbits of the shells. Neutrons have no charge. That's why they call their neutrons. So they call them neutrons. Okay. Then the number of electrons is the same as the number of protons in the neutral atom. Then we have what we call atomic mass. The atomic mass is equal to the number of protons plus the number of neutrons. If you add the protons and neutrons, you get the atomic mass. Okay. And if it's an example here is oxygen. Let's take the oxygen. I have 16 and 18 written on the left side. The top one, 16, there's superscript, is atomic mass, if they write it this way. And then the subscript, the 8, is the atomic number, okay, if they write it this way. Okay. Now, the electrons in the outermost shell are what we call the valence electrons, and they are involved in chemical reactions. Atoms with the same number of valence electrons belong to the same group on the periodic table. I'll show you shortly. And then the other thing is when an atom loses an electron, it becomes positively charged. So let's say you have aluminum. Aluminum, if it loses its outermost electrons, it becomes aluminum 3 plus, and so on. If it gains an electron, then it becomes negative. Okay. So those are some few things that we're going to talk about. When an atom gains an electron, it becomes negative charge, like oxygen. And charged atoms, when you have a charge on it, we refer to them as ions. We call them the ions. Okay. So basically what I'm saying is that if you have the atom, take the atom, it has a nucleus. And the nucleus will have the protons and neutrons. Then the electrons will move in what's called the orbits. So electrons will be somewhere. And this is the orbit. Okay. So orbit is just the path in which you find the electrons. Okay, they move like in a circular path, and that's the orbit. So this is positively charged, this is neutral, and this is negatively charged. So that's a summary of what I have there. Okay, the summary. So let's apply what I have here to some questions that they ask you. So on the table, I've created a little table with some information here. Okay. So the first one is sodium, Na. So sodium at Na, atomic number is 11. So this will be the atomic number. Atomic number is 11, and then that will be the mass number here. So we said that the atomic number, which is 11, is equal to the number of protons. So I have 11 as the number of protons. Now the number of protons, as we said, is the same as the number of electrons. If there's no charge on the element, no charge on it. So it means the electrons would be 11, but there's no charge here. Then the neutrons, we said that if you add the protons to neutrons, you get the atomic number, which is 23. Right? Protons plus neutrons, atomic number. So it means that if I subtract 11 from 23, it gives the number of neutrons. So that is 12. Okay, so the neutrons will be the difference between atomic number and atomic mass. Okay, and that's how I got the 12 here. Okay. Same thing if you work with the oxygen. You have 16 atomic mass, and then the atomic number is 8. Okay, so the atomic number is the same as the number of protons. That's why I have 8 here. 8. The number of electrons is the same as that number of protons. So again, it's 8 because there's no charge on the element. Then the neutrons is a difference between the atomic number and atomic mass. So you have 8 neutrons here. Okay. And if you do the same for carbon, you have 6 electrons, 6, and then the neutrons will be 12 minus 6, and that will be 6. So that's how you identify the protons, electrons, and neutrons. You can be asked to come out with the numbers. They'll give you the structure, and then they'll ask you to find one of them. Okay, that's how they ask the question on this. Okay. Now let's move to the third column here, and that is what's called the electronic configuration. The electronic configuration. Now, when it comes to electronic configuration, we use this system. Let me get rid of this here. So I'm going to draw the atomic structure. Okay. So if you take, let's type the sodium, which is the Na. If you take the nucleus inside the nucleus, now the first orbit usually can take 2 electrons. I hope you all remember that part in your chemistry. Maximum of 2, right? So since sodium is atomic number is 11, you're going to arrange the electrons. So you have 1 electron, 2 in the inner shell. Then you go into the inner shell. Once you have 2 in the inner shell, it's maximum. You can put in more. Now we can put in maximum of 8 in the next shell. So that 1, 2, 3, 4, 5, 6, 7, and 8. So we have how many? 10 now. So 2 innermost, 8, maximum 8, next one, and then 1 more left. So 1 electron will go on the other orbit. So now I have 11 in all. Okay, 11. So this is the electronic configuration. On the test, you're not going to have time to be drawing all this here. So you can use the configuration that I have here, the system that I have here. So the inner shell is 2. So we're going to write 2 for the inner shell. Then column. Then the next orbit, right? How many electrons have next orbit? You have 8 in the next orbit. So we're going to write 8 maximum. And then go into the next shell. And the last one, you have 1 in the outermost shell. So this is the first shell, second, and this is the third orbit. So it's 2, 8, 1. That's how I got this here. You know, a quick way of doing this problem. Okay. Then if you take something like, let's draw something like oxygen. Oxygen atomic number is 8. So if you want to do the same configuration here, then to be 2 in the innermost. So I have 6 more left. So 6 electrons goes in this one. So this will be the configuration for oxygen. 2, 6. Okay. If you take for carbon, atomic number is 6. So the innermost will be 2, and then leaving 4 in the next orbit. So this is for carbon, and so on. Okay. So you don't have to draw all this structure like this to save you time. Okay. All right. Now the electron in the outermost shell, this outermost shell, this is what we call the valence electron. Okay. The outermost shell. So there's a 1 here. So the orbit is called the valence orbit. Okay. Valence. Okay. Electron in the outermost shell. So when they ask you what the valency of, let's say, sodium, so the valency of sodium will be 1. It means how many electrons you have in the outermost shell. So valency here will be equal to 1. Valency of oxygen. Valency of oxygen over here, the valency in the outermost shell, you have 6 electrons. So the valency will be equal to 6. When you come here, what's the valence of carbon? The valence of carbon is 4. The number of electrons in the outermost shell. Okay. Okay. So if you draw for a couple of elements, let's say something like potassium. If you draw for potassium, potassium will have a valence of 1. So because of that, we put sodium and potassium in the same column with the periodic table. If we get a chance to look at the periodic table again, that looks like that. You see that in group 1 will have valence of 1, group 2, valence of 2, 3, valence of 3, and so on. Okay. So all those in group 1 have 1 electron on the outside. All those in group 2, 2 electrons, group 3, 3 electrons, and so on. That's how the periodic table was created based on the valency. And all the chemical reactions just involve the valence electron. They have similar reactions. That's why they group them together. Okay. All right. Now, what happens if you lose the outermost electron? You can lose the outermost electron. because it's easy to lose that because it doesn't need too much energy. So if you take the outermost electron, let's begin with sodium and look at what happens. Now, if I lose the outermost electron in sodium, let's get rid of this here. Okay. Okay. So if I lose this outermost electron, that means this goes off. I'll end up with 2,8, right? 2,8 will be my new configuration. So I have sodium, because it lost one electron, it becomes +1, 1 plus, right? Usually one, we don't write the 1, just plus, just 1. It loses the electron and becomes what we call it's cation. The configuration now will become 2 and 8. 2, 8. Okay. It becomes stable. Okay. So any atom with maximum of 8 on the outside is stable. Okay. They want to come like the noble gases, those in the period 8 group. They all have 8 on the outside. Okay. They want to become stable. So sodium now becomes stable by losing 1 electron. So it becomes plus. So that's what we have in the table here. You lose the outermost electron, they have 2 8s. Okay. So it becomes a cation. Okay. Sodium loses an electron from its outermost shell to become like the noble gas. 8 electrons in the outermost shell makes it stable. Okay. That's what I have in the comments. Okay. Likewise, if you take oxygen, oxygen is 2 6. Oxygen is 2 6. O2, we have 2 6. Now for oxygen, it will take too much energy to lose all the 6 electrons on the outside. So oxygen would rather like to gain electrons. It would like to gain. So this one would like to gain electrons. Okay. So if it gains electrons, it can gain 2 electrons. Then it will become 2 8. So now it's going to become 2 8. It gains 2 electrons to become 2 8. So once it gains 2 electrons, now we have more negatives than positives. So that's why I have 2 negative electron gain, right? So this is what we refer to as the anion. Anion. So electron gain becomes negative, electron loss, it becomes positive. Okay. So the comment I have is oxygen gain 2 electrons to become stable like the noble gas. So adding 2 electrons to this then becomes 8. Okay. So that's how you come up with the configuration, the electronic configuration. Okay. Any questions so far on the electronic configuration? You want to apply that to another concept? Someone has any questions on that? So far, so good?
Yes.
Okay. All right. So with this knowledge, now we can talk about the chemical bonds. So let's look at the screen for that one. The first one we have is called the ionic bond. Ionic bond. So the ionic bond is the bond formed when an atom loses electrons and the other accepts, right? So have sodium. For example, sodium we have 281, and a chlorine, if you take chlorine, is 287. Atomic number is 17, so we have 287. Now sodium has 1 electron on the outside, valence of 1. Chlorine has 7 on the outside, so it has valence of 7. So sodium can lose its atoms electron, which is 1, and give it to the chlorine. When this happens, then you have sodium becoming 2, 8. It's stable. The chlorine gained 1 electron, so it becomes now 8 on the outside. So you can see chlorine is also stable, right? So both are stable. So you have N plus, then chlorine becomes N minus because it gained 1 electron. Then they can combine to form sodium chloride. So this is what we call the ionic bond. Okay. Transfer of electrons from one atom to another to form a bond. That's ionic bond. Okay. Then we have what we call the covalent bond. The covalent bond. This is the bond formed through the sharing. So the key word here is sharing of electrons. They share. You know, they can transfer. They just have to share electrons. So it is formed when you have a non-metal and bonded to another. Okay. Let me go back to the ionic bond and let's make a little note here. Now, the ionic bond here is formed between, if you have a question where you have what we call a metal and bonded to a non-metal, then you can form ionic bond, right? So the periodic table, the metals are towards the left and the non-metals are towards the right side on the periodic table. Okay. So we've seen an element from the right side and an element on the left side, they will form ionic bond. Whilst in covalent, usually it's non-metals that form the bond, non-metals. Okay. So an example would be if you have H2, Cl2, HCl, these are all non-metals. So they will form ionic covalent bonds, Okay. So if you look at what I have here, now hydrogen has 1 electron and then that hydrogen has 1 electron. The only way they can form a bond is to share. So oxygen, this brings its electron, the other one brings an electron, and they share. And then you have H2. The dash line here represents a bond, which is made up of 2 electrons. Every 2 electrons makes a dash to represent that covalent bond. Okay. When you get to chlorine, let's take chlorine for example. Chlorine on the outside, we are looking on the outside electrons only, the valence electrons. You have 7 electrons on the outside. These are 7 on the outside. The only way you can form a bond is to share the electron. They can share this single electron can bond with that. And then you end up with a structure like this. So the 2 electrons form a bond. And that is this dashed line here. That they share the electron. So this is an example of another covalent bond. Okay. And if you take H and chlorine, hydrogen and chlorine, the same thing. Hydrogen can bring this electron and share with 1 of the electrons of chlorine. Now the chlorine will have 8 electrons. It counts 8. The hydrogen now has 2. Once hydrogen has 2, it is stable. It's happy with itself. You know, then they form a covalent bond. So the key word is sharing of electrons. Sharing of electrons to form a bond. Okay. Yeah. Now, even though they are sharing, let's make mention of that here. Now, water, let's look at water. Water is also formed using a covalent bond, right? I'm going to draw the water molecule here. So H with oxygen and hydrogen, that's H2O, right? H2O, water. 2 hydrogens bonded to oxygen like this. Okay. And then even though they form it's formed from a covalent bond sharing, what happens is that oxygen is more powerful than hydrogen. It likes to pull electrons towards itself. So we use the word it's more electrophilic. Oxygen is more electrophilic, so it pulls electrons towards itself. even though they are sharing. It's like stealing from the hydrogen. So at every point in time, you have more electrons on oxygen. So we use the word like delta symbols, delta negative, more electrons on oxygen than hydrogen. The same thing here. So this one has lost electrons. So it's delta positive, a little bit delta positive, be partial, like a math symbol delta means partial. So more negative than hydrogens. It's like pulling electrons towards itself. Okay. So this gives the water what we call a polar nature. So the water becomes polar covalent. Polar covalent, right? Because of the unequal sharing. Okay. So if we go back, I said water is purely ionic. It's neither purely ionic or purely covalent because the electrons are not shared equally. So keyword, not shared equally. So when there's unequal sharing, you have what we call a polar covalent bond. Unequal sharing. You have polar covalent bond, like the water. Okay. When the sharing is equal, then you have a non-polar covalent. Say non-polar covalent, then you have equal sharing. So that's the key takeaway here. Unequal sharing becomes a polar covalent compound. Non-polar covalent, equal sharing. Okay, we can have this also in questions. How can I identify equal sharing? Equal sharing. Let me see if I have examples here for you. All right. Okay, so for equal sharing, examples would be if you have H2, Cl2, O2. The atoms are the same, so they don't cheat each other. They share equally. So if you see molecules like this, then you can say that they are non-polar covalent, non-polar covalent compounds. Okay. Equal sharing. All right, so that is what I have on the screen here. A little explanation to explain the electronegativities. So I said that as a rule, when the difference between electronegativities of two elements is less than 1.2, we assume that the bond between the atoms of this element is covalent. When the difference is larger than 1.8, the bond is assumed to be ionic. Compounds for which the electronegativity difference is between 1.2 and 1.8 are best described as polar or polar covalent, right? These ones are numbers that are assigned to each atom, called the electronegativity numbers. If you take the differences and you get these values, then you can classify them as polar and non-polar. But most of the time on the test, you can use the idea that I get to you. Those on the far left, sorry, on the far right, they are more electronegative than those on the left side. Okay. So that's how you can identify an electron-loving group. Far right, they pull electrons from those on the left side. Right. So with that knowledge, you have what we call the hydrogen bond. Hydrogen bond. Hydrogen bond is a special type of bond. When hydrogen atoms are joined in a polar covalent bond with small atom of high electronegativity, such as oxygen, fluorine, nitrogen, the partial positive charge on the hydrogen is highly concentrated because of its small size. If the hydrogen is close to another oxygen, fluorine, or nitrogen, another molecule, then there's a force of attraction termed a dipole-dipole attraction. This attraction is known as the hydrogen bonding. Hydrogen bond has a very important effect on the properties of water and ice. Hydrogen bonding is also very important in proteins and nucleic acid and therefore in live processes, right? So to use the water molecule again, the hydrogen bond is a special type of bond. If you have hydrogen bonded to something, and you have nitrogen, oxygen, and fluorine somewhere, only these three elements will form what we call the hydrogen bond, special type of bond, right? So if I take the water molecule, H2O, I'll draw that here, oxygen, oxygen. We said that this is slightly negative, this is slightly positive, and this is slightly positive. So this one molecule of water. If I take another molecule of water, I'll have the same thing. So let's say H, water, and another one. A little bit negative, a little bit positive, a little bit delta negative positive here. So that's another molecule of water. So it's also polarized. It's also polar covalent. Now, positive always attracts negative. So the oxygen will interact with the positive hydrogen here. There's an interaction between the two. And this interaction here is what we call the hydrogen bond. The hydrogen bond interaction between the two. So that's what holds the water molecules together, the hydrogen bonding. Okay, I can add one more here. If I have oxygen, hydrogen, positive, positive, negative. This will interact with that. And then that holds a bond together. So those are hydrogen bonds and so on. Okay. Okay. So the same will happen if you have nitrogen somewhere, like nitrogen. So if I have NH2, NH3, sorry, you're going to have hydrogen bond because the nitrogen steals from the hydrogen. Nitrogen will always pull from hydrogen like this. This is slightly positive, slightly positive, slightly positive, and this is negative. So this molecule will look like this. If I have another molecule, the same thing, nitrogen, hydrogen, 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 positive, 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 and then negative. So the hydrogen interacts, positive interacts with negative, and they have a bond. So that will be a hydrogen bond in between. Okay. So remember, you should have only these three elements, NOF, nitrogen, oxygen, fluorine, with hydrogen attached to it before you can have a hydrogen bond, special type of bonding. Okay. All right, great. So this brings us to the end of the chemistry part, some of the chemistry I want to tackle today. The atoms and the bonding for now, and then we'll continue with the other parts. Yeah. So let me pause any questions so far on the chemistry. The first concept of chemistry, nitrogen, atoms and the bonding. Let's take some few questions before we continue. Any questions on this so far? The general concepts. Anybody has a question?
No.
So far, so good. Okay.
Yes.
All right. All right. So let's continue with the next one then. Those one will be faster, and then we'll look at some questions. Okay. Okay. Right. So this will be good to go now. We're on track. All right. So the next one is the eye. The eye, you have to know that also inside out. Every year when they come back, I hear people tell, oh, the eye was there, the eye diagram was there. So hopefully when you also go in, you may see the eye. You know, so the eye, I always stress on it, know it very well. And there's some few things that I'm going to highlight. This one shows you all the different parts of the eye, but it's not all that you need to know for this test and this basically. So remember, that you have the conjunctiva that covers the surface of the eye like this. So it's a thin protective layer that covers the surface of the eye, you know, and the lining of the eye, in the line of the eyelids. That's the conjunctiva, the one covering the blue one. And then the blue here is the cornea, the cornea here. So the cornea is a clear area of this, what we call the sclera. The sclera is the outer coat, which helps to bend light into the retina, right? So the function is bending of light, right? The other side, the sclera is the back side, which is not transparent. Okay. The front part of the sclera is the cornea. Okay. Then the iris, the iris here is a colored part, right? There's a colored sheet of muscle which controls the size of the pupil, you know. So it controls how much light enters the eye. So that's the iris, control the size of pupil. So if somebody moves from a dark room to a bright room, the pupil would constrict, becomes narrow, so that it will reduce the amount of light entering. It's a way of protecting the eye. If a person moves from, let's say, a bright room to a dark room, the pupil will dilate, so that more light, you need more light to see. So it will dilate, so the person can see well, you know, and that is controlled by the iris, the muscles of the iris. Okay, so know the function of the iris. That's very important to know this one, the function of the iris. Okay, then the lens, the function of the lens. The lens is for focusing. So no, the lens is for focusing. Another name for the focusing is you like to use the word accommodation. Accommodation. So see, accommodation is a word focusing. That's the function of the lens. Okay, so the object is far, it will change the structure to be able to focus the image on the retina. Okay. Then the other thing that I would like you to remember, and that this is the retina. Where's the retina? Okay, the retina is here. The retina, they like asking questions about that also, the retina. Okay. So remember, the retina is where the image is formed. Okay. And I would like to mention that it has two what they call the photopigments. One is called the rods, and the other one is called the cones. They like the rods and cones. I'm going to talk about that shortly. I think I have some notes on that. So let me see if I have that. So remember the rods and cones in retina. Remember the function of the lens, the iris. Remember that. Those are the important things here. Then there's a fluid in the back behind the lens that is called the vitreous humor. That's vitreous humor. It's like a jelly. It's more like a jelly-like substance, you know. And then the front part of the lens over here that they call anterior chamber, you have the liquid. It's more liquid of known as the aqueous humor. That's the aqueous humor here. Yeah, like it just helps to maintain the structure of the eye. Okay. When the aqueous humor is too much, then the pressure in the eye increases, and that's what we call glaucoma. I'm sure you've heard of glaucoma. It's increased pressure in the eye, okay, because of too much fluid, aqueous fluid. Okay. And then the other thing you should remember is the optic nerve. The optic nerve, that's a nerve that transmits the impulse to the brain, right? Transmits the impulse to the brain. So that's function. I think I have a summary. I put down like a summary somewhere. Let's see. Yeah, it's here. Okay, I tried to put a lot of information to make it a lot visual for you. All right, so this is the structure of the eye. Again, the functions, I put them together here. So what they do is that they can give you this diagram, and then the boxes will be empty. Maybe they can label box 1, 2, 3, and you should be able to identify the parts and then the function, parts and function. Right. So when you're studying, think about this diagram here. And these are the key points that I'm talking about. The key points, function of the lens, iris, pupil, and the retinal rods and cones. Now note that the rods are needed for night vision. Night vision for the rods. Let me highlight that. You need the rods for night vision, since they are very sensitive to light. So that's the function of the rods, night vision. So see, night vision goes rods. Then they also help you to distinguish between black and white colors, black and white. So night vision and black and white. While the cones, they are needed for daytime vision. They are less sensitive to light, so you need a lot of light to stimulate them. So they are needed for daytime vision and also for distinguishing between colors. Distinguish between colors and daytime vision. Yeah. So if somebody has like problem with colors, like the person is colorblind, it means that there's a problem with the cone. The cone has a problem, right? So that's why they can't distinguish between the colors. That makes them colorblind cones. So those are two things that you should remember. If you forget everything about the eye, don't forget the rod and the cones in the retina. Rods and cones in the retina. Very important question that they like to ask. Okay. All right. Okay. So we'll be seeing that in questions shortly. Okay. Rods and cones. Then the other thing you have to know about the ear and its function. The ear structure. Usually they're not going to give you the diagram for that, but you should know about these few things here. Remember that the ear has three parts. The outer ear, so up to, if I draw up to here, this is the outer ear. From the external, which is this called the pinna, to the tympanic membrane, that's the outer ear. They have the middle ear from the tympanic membrane to this structure. So this is the inner ear here. Tympanic membrane to this cochlea, this is called the cochlea. And then from here, there is the, no, I made an error. This is the middle ear. This is middle ear, and the inner ear is here. The inner ear. Okay, the cochlea is in the inner ear. Okay. The few things I want you to note here, it's the function of the cochlea. The cochlea, if you see the cochlea in the question, remember the cochlea function is for hearing. For hearing, cochlea goes to hearing. Those are the key points you should remember. So hearing, anything of hearing, think of the cochlea. Hearing, cochlea. Okay. So if somebody has a problem hearing, think of the cochlea. Then they have what they call the vestibule. It has two parts, the vestibule, cochlea and the vestibule. The vestibular portion that one is associated with balancing. Balancing. Okay, so the cochlea and it has a vestibule to cut across somewhere here. The vestibule somewhere here for balancing. Cochlea for hearing. Okay. Then you have these semicircular canals. This here, semicircular canals. There are three of them. They are arranged at right angles. So it's like X, Y, and Z planes, right angles. Okay, X, Y, Z. If you take like the wall, four corners of the three corners of the wall, 1, 2, 3, right? Vertical, horizontal, in the X and Y planes. So that's 1, 2, and 3. Okay. The semicircular canals, the function is also for balancing. Helps you to balance. So all the planes are covered, the three-dimensional planes. If you tilt your head in any direction, it will send impulse to the brain through the vestibular nerve to the brain, and then you balance yourself. Okay, so those are the three things that you should remember here. Okay, and then the last one that I want to mention for the ear is the eustachian tube. So the eustachian tube, remember that one is for balancing the pressure in the ear. Balancing ear pressure. Balancing the ear pressure in the middle ear. Okay. So this connects to the pharynx, the mouth. All right. So if you're sitting in the plane and the pressure drops, you feel that funny feeling in the ear. Is because of air ration, because of collapse. Sorry, the eustachian tube collapses if the pressure is too much. So in order to balance the pressure to protect the ear, you have to swallow something. Once you swallow something or you chew on something, then it opens up and the pressure balances between the atmosphere and then the middle ear. You know, so it's for balancing. Then the last piece are these structures in the middle ear. You have these structures here. This structure here, I hope you can all see that, called the ossicles. You call it the ossicle. They're like bony structures, 3 bony structures. The first one is called the malleus. You have the next one called the incus, and then the third one is called the stapes. The malleus, incus, and stapes. So together we call them the ossicles. Ossicles. So the ossicles are the malleus, incus, and then the stapes. Okay. Those are like a bony structure, small bones. And the function, they're there to increase the intensity of the vibration. Because the sound vibrates the tympanic membrane. That's the eardrum. This is the eardrum. So it balances, it vibrates, and then this increases the vibration, the bones, they increase the vibration. And then there's fluid within the cochlea, which also vibrates. It's like stepping on the paddle of a drum. It vibrates it, and the vibration generates an impulse within the cochlea that is transmitted along the cochlea. It'll be complex when you cut through this, but we're not going to go through that. Just know these basic information for the ear. Okay. Cochlea for hearing, semicircular canal for balancing, eustachian tube balancing the pressure in the middle ear, and then the function of the ossicles, the 3 bony structures that increase the vibration. All right, so I have a little summary here. What did I say? I have them here. Okay. There's some few questions. I said, what will happen if the cochlea is damaged? What will happen if the semicircular canals are damaged? So these application questions that they ask you. So always don't, I always tell people, don't think they're going to ask you, what's the function of this? What's the function of that? You know, they can ask scenario questions or application questions like what I have here. You know, what happens when the cochlea is damaged? To test you if you know the function of the cochlea. And the function is for hearing, so it can be affected. What happens when the semicircular canals are damaged? The function of semicircular canals is for balancing. So if they're damaged, then the person will not be able to balance himself or herself. Okay, and so on. Okay, application questions like that. Okay, yeah. Great. And then the last overview that I want us to look at is that of the brain. A quick overview of the brain. Okay. So if you look at the brain, you have the big parts here, the bulk of it, all this here is a cerebrum. And then we have the cerebellum. Then we have the brainstem, which is here, called the brainstem. Okay. We're going to talk about the functions quickly. So the brainstem is made up of all these pons, medulla oblongata, and so on. Now, so basically, if you take the cerebrum is for like higher thinking processes. I think I have another slide that discusses all that. So the brainstem, sorry, the cerebrum is more for like higher thinking processes. They have different parts: frontal, parietal, occipital, temporal, and so on. Those who have done anatomy physiology, right? And the frontal part is mostly for thinking, initiating movements, your behavior, memory, speaking, and so on, right? And the parietal part is more like sensation. How when you touch something, you going to tell whether it's soft, and so on, hard, and etc. You know your left from right, sensation and reading. And the occipital part is for vision. So if that is affected, vision is also affected. And then the temporal part, mainly for hearing and understanding language, and also behavior and memory. Okay. Then the brainstem, which is made up of the mid, it has what we call the midbrain, the pons, and the medulla oblongata, which I just showed you from before. The function is for the brainstem, especially the medulla oblongata, controls breathing, blood pressure, heart rate, and swallowing, right? So main function of the medulla oblongata is mainly cardiorespiratory, as a cardiorespiratory function. Okay. So I asked the question, why is the medulla oblongata very important? It's important because it is the cardiorespiratory center. It's a cardiorespiratory center. Very important. If you forget everything, at least function of the medulla oblongata, don't forget that. Cardiorespiratory center. It controls the heart rate and the respiratory rate. Controls heart rate. Controls respiratory rate. Okay. So that is the function of the medulla oblongata. Okay. So this is again very important as future nurses, because if somebody has what's called a whiplash injury, like there's a car accident, the person's neck goes like this, whiplash injury, you can fracture the first cervical bone and the second cervical bone. There's something called the dens, if you remember for anatomy. The first cervical bone has a projection on the bone like this. That's the second bone, and then the first one goes around it, like a ring around that one. So if someone has an injury, this bone can fracture, and the brainstem is somewhere here. The brainstem is passing through your brain. So if it fractures, it can compress on the midbrain, the brainstem. So if you compress on brainstem, it means that the medulla oblongata will be compressed. And once it's compressed, the heart will stop, and then the respiration will also stop, and then there's a problem. So that's why when someone has a whiplash injury, when the paramedics come, they put a collar around the neck to stabilize the neck before they lift the person up. Okay, if you just go and lift the person up and you bend the neck, you may rather kill the person, because you'll be compressing on the medulla oblongata more. Yeah. So you stabilize the neck first before you lift up the person. Okay. All right. So this is a quick overview of the function. And then one more thing, remember the cerebellum? The cerebellum, the function of the cerebellum Again, balancing. Keyword, balancing and coordination. So keyword is balancing. So cerebellum for balancing. Okay. So those are three key things. Cerebrum, higher thinking processes, understanding, personality, and so on. Hearing. Whilst you get a brainstem, medulla oblongata, very important structure there. And then the cerebrum, balancing. Okay, so the cerebrum works with the semicircular canals in the ear to help in balancing. Okay. So that's the quick overview of the brain. Okay. And then the last piece that I want to touch on the science for today is the reflex arc. The reflex arc. So remember, the reflex arc basically is not under your control, right? Not under your control. So what we call the autonomous. It's automatic. Okay. So if somebody steps on a sharp object, the signal is sent through what we call the efferent neuron. The efferent neuron. So know the reflex arc. It starts with the efferent neuron. Another name for that is a sensory. Sometimes they ask you about the path. Sensory neuron. That is this. And then the message goes to a spinal cord. So this is a spinal cord. It occurs at the level of the spinal cord. That's why you have no control over it. Then there's a second neuron known as the interneuron. So this goes to the interneuron. It's a short neuron. And then from the interneuron, the message is interpreted quickly and the response is sent through the efferent neuron. The efferent neuron. The efferent neuron is also known as the motor neuron. And then the message goes to the muscle. If it's your leg, then you're going to lift up your leg quickly. And so if the arm, you're going to move it quickly. So it's a quick response. So from efferent to interneuron, then to the efferent neuron. Okay, that's the pathway. Okay, and then the response is generated. Okay, so this will occur quickly before you have some message going up to the brain. That will be the second response to tell you something has happened. Okay, by initial response, you have no control over it. You lift your leg before you think. Okay. All right, so this is a brief overview of the things I want to discuss on the science part for today. Okay. So we're going to answer a few questions before we do the math part. I'll introduce you to the math part. Let me pause for a second. Any questions so far before we go to the book? See how you can apply to questions. I'm sure that's what you're all waiting for. How do you apply to questions in the book? So any questions so far on all that I've said for today? I think that the notes I have is detailed enough. Any questions? Let me hear your responses.
No.
No.
Yes, I can see you clear. Everybody, no?
No.
So
No.
far, is everything understandable?
Yeah.
Yes.
Okay, thank you. Right. So we're doing the best we can to make sure everybody gets something right. Right. So let's use the next few minutes to, before we switch over, let's go to a textbook. If you have a textbook, let's look at some questions, how we can apply today's knowledge. So this way, I'll need a lot of interactions from you to tell me the answers so I can test your understanding as we go on. So if you can open your textbook, let's start from, I'll be jumping from question to question based on then from what do you call it, the schedule, the related questions for today. So we'll go from page to page. Let's look at page 218. If you have a textbook, 218. You can open to that page. If you don't have it today, I'll read out the questions for you to hear. Okay. Let's look at number 3. So I'd like to hear some responses from you as we go through. The first one says, the spaces between neurons are called A, synapses, B, dendrites, C, interneurons, and D, cell gaps.
A.
A, synapses, that's correct. Synapses, the spaces, synapses. Good. All right, so if you're not happy with the answer, you can just let us know and then we'll discuss it further. But
Professor, what page is it? Because
218.
218.
Yeah, 218.
The question that I have is different. It says an organism with chloroplast in its cell is probably.
Put your hand in the back.
Yeah,
That's
that's the same question I have.
the same question you have.
Yep.
Which is a different edition?
It's the
I
third
have
edition, the one that was given the link to.
Yeah, the 3rd edition.
Well, my number is different, so I don't know. Probably they change something because I have this 3rd edition too. But maybe they modify something because the one you're saying is number 1 for me.
Question number 3, not number 1.
Are you on number 3?
Okay, yes, I see the question on number 3 now.
Yeah.
It was
Okay, good. Yeah. So we look at the questions related for like what we've done today. So number 3, that's A. Okay, good. We're all there now. Okay. Number 4. Let's look at number 4. Which group of chemicals is not? So the keyword is not normally found in most living things. A is carbohydrates, B, proteins, C, silicates, and then D, nucleic acids. So based on today's knowledge, which one is not found in most living things? Which one?
C.
D.
C.
Somebody said B, somebody said D.
I would say C, like a cat.
Yes, the answer is C. The answer is C. You see, remember we mentioned carbohydrates in the lesson. We mentioned proteins and we mentioned nucleic acids. Nucleic acids are the nucleotides, right? So silicate is not, the keyword is not found. Silicate is sand, right? Sand. That's what makes up sand. So we don't have sand in living organisms. So C, silicates, we don't have that. Okay, good. Let's look at number 8 on page 219, the next page, number 8. It says, which of the following organelles is not involved in protein synthesis? Not involved in protein synthesis. A, ribosome. B, mitochondrion. C, rough endoplasmic reticulum. And then D, nucleus. So which ones are involved in protein synthesis?
B.
Oh, it says not. Yeah, so B, correct. So protein synthesis, let me write that here. A quick, what is it? Okay, let's see.
I would say D.
The answer is B. It's not. The question says not. All right, so basically protein synthesis, this is what happens. I was going to talk about that another day fully, but you start with the DNA and then you transcribe that. Transcription. Then you go to RNA. You form what you call the messenger RNA, mRNA. I hope you all remember that part. And then the mRNA is what is translated. We use the word translated. It's translated to protein. Okay, that's how protein synthesis occurs. Protein synthesis. And we know that the DNA is in the nucleus. Remember this one, the nucleus? DNA, the genetic material in the nucleus, right? So the nucleus is involved. Now, the rough, once the mRNA is produced, you have other structures called tRNA. There's something called tRNA here. So this is the strand. The ribosome does the reading. The ribosome that does the reading is called tRNA. That reads the code. No. This is R, sorry. It's rRNA. That reads the code. And then you have one that brings the amino acid in, a shape like this. The shape looks like this. It carries the amino acid. And this is called a tRNA, transfer RNA. Okay, so rRNA reads the code. This one does the reading. Reads the code. It does the reading. Okay, right. And then the rough endoplasmic reticulum is what carries the protein that is formed. So it transfers protein. If the first part of the lesson, on this, you see rough endoplasmic reticulum carries the protein or the substance within the cell, right? So if you look at all this, mitochondria is not involved. Energy is not involved in here. Okay, mitochondria is not involved. So ribosome is involved, reads the code and the messenger RNA. Endoplasmic reticulum transfers the protein that is formed. And the nucleus, we have the DNA in the nucleus. That's why B is not involved. All right.
Yeah, I was confused because I checked the answer at the back of the book and it says the correct answer is D.
It's B, yeah.
D as a
It said
direct.
B.
D.
Oh no, it should be B.
Yeah, but at the end of the book is D. That way I was like,
Yeah, there's a few errors that appear in the book. I've seen
Thank
some of them. All right, good. Great. So number 12, look at number 12 on next page. Page 220. Number 12. To be absorbed by cells, proteins must be changed to A, amino acid, B, sucrose, C, fatty acids, and then D, this glycerol. So we are breaking down protein. In other words, what's the basic unit?
A, amino acids.
Correct, amino acids. So the basic unit of protein is amino acid. Okay, great. Amino acid. Okay, number 13. A neuron that transmits impulses from the receptors to the spinal cord is called, so now we think of the reflex arc here. A,
Sensory neuron.
motor neuron. B, an associative neuron. C, an interneuron. And a D, sensory neuron.
D.
D, correct. So it's bringing from the receptor to the, so it's right there on the screen too. For the receptor, this is a receptor that is like the skin receiving, and then it brings, so is the sensory neuron, correct. Another word is the efferent neuron. Efferent neuron, good. So next time they can change it around and then use other ones. So good to know the diagram. Okay, let's look for another one that is related to today's lesson. Okay. All right, page 223. 223. Number 27 and 28. We look at 27 and 28. Are we there? Okay, let's go. Which of these foods is a good source of protein? So this is like use your general knowledge. Which of these foods is a good source of protein? A, nuts. B, cooked oatmeal. C, honey. And then D, raisins. Good source of protein. So you can use your general knowledge.
A, nuts.
A, nuts, correct. A, nuts, right? So B, C, D, what was it? What category of food would you put that? Which category? The cooked oatmeal, honey, and raisins.
I'm sorry, can you repeat the question?
Oh, my question is, which category will you classify B, C, D? Oatmeal, honey, and raisins. Where will you put them? And the food
Carbohydrates?
Carbohydrates, yeah. They are high carbohydrates. Good. 28. When oxygen goes from air in the lungs into the blood, it does so by, so they're asking about the movements now. A, catalysis. B, diffusion. C, active transport. And then D, osmosis.
Diffusion?
Yeah, it's simple diffusion. So one of the examples that I gave, simple diffusion. Simple, no energy is needed. Simple diffusion from high to low, high to low. All right, great. Okay, another question. Page 225. Page 225. Number 39. It says, which of these nitrogenous bases can be found in RNA but not in DNA? RNA but not in DNA. A, uracil. B, adenine. C, thymine. And then D, guanine. Uracil, adenine, thymine, and guanine.
What question is this on 229?
25. 225.
What question?
Number 39.
39.
Thank you.
39.
A, uracil.
Yeah, uracil. So uracil in RNA, but not in DNA. Correct. All right, so far so good. Page 226. 226. Number 45. Two six six, number 45. It says, which diagram represents a non-polar covalent bond? Non-polar covalent. So there should be sharing somewhere, and then it should have equal sharing, non-polar covalent. So the diagram A, B, C, D, which one will you choose? Anybody? So
What number is that again?
45. Page 226, number 45.
It's A.
A.
It's A, correct, because it's Cl2, right? Cl2. So it's equal sharing. We saw that. I think I drew that as well. So equal sharing. That's correct. B and D, those are polar covalent. B and D, polar covalent. Okay. And then C will be ionic. Made of ions, ionic bond. Okay. So answer is A, chlorine, chlorine, equal sharing. Okay, 46. What would answer for 46? Certain seaweeds accumulate iodine in concentrations as much as 1,000,000 times greater than that of the surrounding ocean. How must this intake be accomplished? So this is one of the movements of substance question. Osmosis, A. B, diffusion. C, active transport. D, passive transport.
Osmosis.
Okay, let's look at the keywords again. It's concentrated and keeps taking in more. It's 1,000,000 times greater than that of the surrounding. Accumulated iodine, 1,000,000 times. How can they achieve that? It's already high, but it's taking more.
D, active.
It's active transport. Active transport. Okay. More inside, but still taking in and accumulating. So that you can achieve that by active transport. Energy is needed for that. Okay, good. That's another one here. Let's see. Page 238. 238. Number 38. No, let's leave 38. We'll come back to that another time. Number 39 and 40.
What page is this on?
238. 238. 238, number 39, and number 40. So, which 39? Which laboratory reagent turns starch blue-black? Which laboratory reagent turns starch blue-black? A, phenolphthalein. B, Benedict solution. C, nitric acid. And then D, Lugol solution.
D.
D.
D, correct. So that's a test for starch, Lugol solution, or iodine test. So next time they will put iodine there. You know. Okay, what is Benedict solution used for? What do you use to test for? Benedict solution.
For simple sugars?
Yeah, simple sugars, right? It becomes, you have the brick red color when you boil it with simple sugars. All right, good. So learn the test for the substances. So that next time you can.
Wait, so what was 39, Professor?
D, the Lugol solution. Yes.
Okay.
Remember the other name for iodine test, iodine tincture, we call it iodine alcohol? Lugol.
Okay.
Yeah, okay. All right, number 40. What types of body cells are most directly involved when a person is walking, when a person walks uphill? Smooth muscle cells, B, subcutaneous cells, C, striated muscle cells, and D, epithelial cells. So which cells for movement of bony parts?
C.
C, correct. Striated muscle, the same as the skeletal muscle. You're moving the bony parts. Skeletal muscle, correct. Okay. Number 41. It says, when red blood coloring is added to a beaker of water, the coloring slowly spreads until the water is all color pink. This is best explained by the process of A, peristalsis, B, diffusion, C, active transport, and then D, osmosis.
D.
D.
Take your time. Someone said B, right? Yes.
D.
D.
B is correct. B. If you said B, that's correct. So this is an example of simple diffusion. I gave this example. You put a dye in water, it spreads through the water. So that is diffusion. The molecule is concentrated and then spreads out to less concentrated. Okay. So it's B. See, there's no membrane. If there's a membrane, then you think about osmosis, but there's no membrane around that the red fluid, red dye. It's diffusion, B. Okay. Now 45. 45. When looking through a microscope, a student observes centrosomes in a group of cells. The cells are most likely from A, the root of a bean plant, B, leaf of a moss, C, skin of a mouse, and then D, stem of a fern. So where do you find centrosomes? Centrosomes. It's in the first part of the nose too. Centrosomes. Animals or
B.
say it again. Which one? Which one?
B. B.
You said B. Let's go back again. Now, centrosomes are found only in animals. Centrosomes are found only in animals. I think it's one of the notes.
I think it's C.
Yes, it's C. It's C. Because centrosomes, you find it when the cell is dividing, the centrosomes form what we call the spindles that pull the chromosomes apart. It's only found in animals. So centrosomes. So look for animal, answer here, answer with animal in it, which is C, skin of a mouse. That's where you find it. Skin, because that's the animal in there. Yeah. Okay, good so far. Okay, let's take one more here. Okay, page 241. 241. Number 54. I didn't mention this, but we can talk about that now. 54 says 2 atoms are isotopes if they have the same atomic number, but a different number of A, neutrons, B, mesons, C, electrons, and then D, protons.
No.
So isotopes. What are isotopes?
A.
A, correct. Neutrons, correct. So for those who have forgotten, let's write down a little bit something here. So when I say isotope, they are atoms with the same atomic numbers, but the masses are different masses. Okay. So example, we have carbon. We have carbon 6 that weighs 12. And you have another carbon 6, but the weight is 14. So these are isotopes of each other. Another example, oxygen. You have oxygen, atomic number is 8. One weighs 16. And there's another oxygen, atomic number 8, but it weighs 18. So these are isotopes of each other. Same atomic numbers, but different masses. But remember, we also said that the atomic number is the same as protons. So this is like saying protons. So we can also say that atoms with the same protons. So these are atomic number is the same as protons. And the mass, if they define the masses, it means they define the number of neutrons, right? Different masses or different number of neutrons. But that's how they define the mass. So that's why A is correct. Okay. And then the question that I gave before, number 56, that is a Caracas question. 56. If a plain cracker is chewed slowly, after a while, it begins to taste more, and I give the answer here on. Salty, A, salty, B, sweet, C, bitter, and then D, bland. Yeah. So we chew it slowly.
D.
becomes sweet, right? B. Because there's an enzyme in the mouth called amylase that breaks down this cracker starch, breaks it down into simple sugars. All right. Let's
I'm sorry,
try
so what's the answer for that one?
sweet. It becomes sweet. The cracker is starch. So if you break it down in the mouth, there's an enzyme that breaks it down in the mouth to simple sugars. And sugar tastes sweet. All right, so let's look at, I think, the last one for this for now. 58. Think about 58. Mutation is change in the base, the nitrogen base in the DNA. So say the first step by which a mutation may occur is a change in A, location of a nitrogen base in DNA. B, messenger
Professor,
RNA.
what number is this?
58. 241, number 58.
Okay.
So the first step by which mutation may occur is a change in the location of the nitrogen base in DNA. B, messenger RNA. C, transfer RNA. And D, amino acid in a protein synthesis. So where would the first change occur? I mentioned that already.
C.
No, I just give the answer out.
Is it A?
A. It's a location of a nitrogen base in a DNA. Okay, mutation means you change one of the nitrogen base. Okay. Remember, if you do protein synthesis, remember that 3 of the bases is called a codon. If you remember protein synthesis, you have codon. So every 3, let's say, have 1, 2, 3, 4, 5, 6. So let's say C, G, C. This is called a codon. And then it's a code for a particular amino acid. It's a code for amino acid. Okay. There may be another A, U, C, another amino acid, and so on. Codons. Okay. So if you change one of the bases, if I replace it for a different base, it will give a different amino acid. Sometimes it may not change. There are some amino acids who have the same codes, different codes for one amino acid. But mutation is knocking off one of, let's say this is knocked off and replaced with another one. Let's say U. You've changed the code. You can see UC. And that will be a different amino acid and so on. Okay. So the change begins in the DNA because the DNA has to copy it to the RNA. So anything that happens in the DNA is going to affect the RNA. Okay. So that's why it's A. It's A. All right, so let's look at maybe one more question here, and then we'll switch for today. Page 242. Page 242. But maybe two other questions, we'll look at that. Number 1. The diffusion of water through a semipermeable membrane is called A, pilocytosis, B, osmosis, C, active transport, and D, transpiration tension.
B.
B, correct. So see, the key word is the semi-permeable membrane. So it's osmosis. That's correct, osmosis. Okay. I saw 2 more questions here, and then we'll round up, I guess, for science. Page 244. Page 244. Number 16, 17, and 18. 16, 17, 18. 16. Which of the following would be classified as an example of a connective tissue? A, striated muscle. B, epidermis. C, nerve. And D, tendon. Connective tissue.
The tendon.
Tendon, correct. So tendon is a connective tissue. So remember I said know the examples of the connective tissue. Bone, blood, tendon, ligaments, adipose tissue or the fat tissue, the connective tissue proper. Right, those are all connective tissues. Right, great. Then number 17, now this is a diagram. They give a diagram and then it's labeled Z. It's part of the cell structure. It says the following diagram shows the endoplasmic reticulum of a cell as seen under the electron microscope. The structures labeled Z are the site of protein synthesis. Z represents A, mitochondria, B, ribosomes, C, chromosomes, and D, centrosomes. So which ones are labeled, those small dots?
Ribosomes.
Ribosomes, correct, ribosomes. So ribosomes, the function is for protein synthesis, ribosomes, right? So again, have the picture of the cell in your mind. You like the cell a lot. Ribosomes. Okay, number 18. The entry and exit of materials in the animal cell are regulated by which of these cell organelles? A, nucleolus, B, cell wall, C, endoplasmic reticulum, and then D, plasma membrane. So what regulates materials entering and leaving the cell?
Plasma membrane.
Plasma membrane, yes, plasma membrane. Yes, plasma membrane is selectively permeable. Okay. Plasma membrane, good. And then number 20, which of the following substances are end product of protein digestion? So if you break down protein, what do you get? A, amino acid, B, lipid, C, fatty acids, and D, cholesterol.
A.
A.
A, yeah, amino acid, the basic units. Good. Okay. All right, so we'll pause here for the science today. So when you go back, just have a look at the questions again, based on the last column. I believe you should be able to answer the questions on the last column of the syllabus. Okay. Okay. All right. I saw one more question somewhere, but I want us to move on. Okay, let's do one more question that I just saw before we move on, and then we page 250. 250. Number 51. Page 250, number 51. It says, a cell is placed in a drop of distilled water. The cell swells and bursts. The most likely cause of this reaction is that, then A, they said active transport of salt occurs. B, water moves from an area of low particle concentration to an area of higher particle concentration. C, water moves from an area of high particle concentration to an area of low particle concentration. And then D, salt from distilled water enter the cell. So what do you think happened here?
D.
The salt don't move. The membrane is semi-permeable membrane, so it doesn't allow salt to move.
Is this C?
C.
Water moves from area of high particle concentration. So the particle is what? High particle concentration. So that is C. Yep. So it's concentrated. The water is where you have more water. That's a concentrated particle to the low, because the salt solution has low water particles in it. Okay. So that's osmosis. The high particle concentration, you talk about the water. So the water is the particle you're talking about, not the salt. Be careful with this question. to an area of low particle concentration. So where you have less water, so that is C. Good. So these are some of the questions that for today you should be thinking about. Okay, great. Anybody has any questions? I want us to start with the math today too. At least have an idea of the math. Okay. Now, okay, let's go to the math part. Today for the math, I want us to start with math test number 1. That's the approach I want to use now. Instead of talking about going over each topic, it will be better for us to look at problems, since you may know some of them already. Yeah. So as we encounter problems, then we'll discuss them so that we're going to complete the math sessions as we go on. We'll cover everything. So page 104, let's do page 104. I believe you're allowed to use calculator now, these days. So at least your life has made it a lot easier. In the past, calculator was not allowed. So you can now use it.
Do they provide the calculators, or we have to bring our own?
It should pop up on the screen as well, but I think some places they allow you to come with simple calculator.
You can use the computer
Very simple
itself,
calculator.
Okay,
but
thank
you can. Oh yeah, you have to bring in your own basic calculator as
Your
well.
basic, yeah. Okay, thank you for that. So let's go to page 104 and then go through the questions. If it's easy, we'll just skip that. I know some of them will be easy for you to do. So if calculator work, we'll skip. So let's try number 1, 2, 3, and 4. Let's try that in about, let's take about 3 minutes to look at that, and then we'll discuss them. 1, 3.
I'm sorry, what page are we on?
104.
104, thank you.
Yes, 104. Number 1, 2, 3, and 4. Let's take about 3 minutes to look at them. Try them yourself first, and we'll discuss. Well, I think someone said she has a PDF of the book. So if you don't have it, she's willing to share with you.
Are we doing only 1 to 4 or more?
If you finish, you can move ahead to do more.
Okay.
Yes, just want to give time for everybody to, like, if you work fast, you can keep going. Okay. Okay, so let's come back and discuss 1 to 4, then you can continue from there. Okay. So I believe for number 1, you should all be able to have that since calculator is allowed.
A.
Yeah, so we'll skip number 1. Your calculator, you had A. Okay. Number 2.
D.
D. Okay, how do you do number 2? Let's share number 2. How do you do number 2?
So there is a question, how many times will it beat in a minute? 1 minute. 1 minute is 60 seconds. So I did like 20 times is in 15 seconds. So X is in 60 seconds. And then
Okay. So what I'm hearing you say is that you use like proportion.
yes.
Well, okay, you can just an easier way. There's 4 parts, so it's
Mhm.
15 seconds, right? So it's, if you just do it 60 times, divided by 15, that's 4. So 20 times 4, right? You get 80.
Okay, right.
That's what I did.
Good. All right. So you use just like a general arithmetic process. That's good. So whichever way helps you on a test, that is fine. If you can identify that you have 15 seconds in 1 minute, then you just multiply this by 4. Okay. But if you can see that, then this is where I'll introduce you to proportion. You can always use proportion for a lot of questions in the book. You can use proportion.
Yeah, that's why I like
A
it.
lot of questions, almost like 80% of the problems will be proportion. So I'm just going to use this to illustrate proportion to you, though you don't need to use it for this question. All right, so the proportion, remember, proportion, we'll be using that a lot for most questions. So you have this beating 20 times in 15 seconds. So I can say 20 heartbeats in 15 seconds. Okay. So set up the same ratio on the other side. So this heartbeat to seconds. So you need to have heartbeats here, and then this should be seconds. Okay. You've been given 1 minute, so we can use the minute. we have to change to seconds. So 1 minute, we know that this is 60 seconds. So this is 60 seconds. We don't know the heartbeat, so we can use X. So this is a proportion. 15 over 20 is equal to X over 60. Then we can use a cross product, cross multiply. So 15 times X is equal to 60 times 20. Right? And then you can divide the left side by 15, divide the right side by 15. This will cross out, so whatever you get to be the X. Okay. Then you can use a calculator to work numerator and then divide by the denominator. Okay. But I like to cross out. So if I cross out, I know 15 here is 1, 15 here, I reduce to lowest times, 4, and then this is 80. Okay. So that's the proportion approach. Okay. So set up the ratio and set them equal to each other. We'll be using that a lot. That's why I'm illustrating with this one. Okay, good. So you had D, which is correct. Okay. All right. For number three, you can use your calculator since you're allowed to use it.
I got C.
C, okay. Let's see. Everybody had that with the calculator. Make sure you know how to use the calculator very well too. Okay. All right. So she has C. But if you don't have calculator, what do you do without if you don't have calculator? You can multiply without calculator. You have to multiply the numbers by without the decimals, right? And then you move the decimal places. Everybody's okay with that? Number three, you can just use a calculator for that. Okay, so let's look at number 4 then. Number 4. Number 4, let's review fractions. Let's use this to review fractions. So remember, when you're working with fractions, addition and subtraction fractions, the key point that you need is LCD. Addition, subtraction. You need LCD. All right, LCD. Okay. So if you have 5 over 12 minus 2 over 3. Before you can subtract, we need to find the LCD. So write each fraction of LCD. So this will be 12. LCD here will be 12. Between 3 and 12 is 12. So we're going to ask ourselves, what was done to this 12 to get this 12 here, to get the LCD of 12? This is times 1. So we'll multiply the numerator also by 1. So that would be 5, right? How do I move from 3 to get 12? I have to multiply this by 4 to get 12. Whatever I do to the denominator, do the same to the numerator. I have to multiply this also by 4. So that gives me 8. All right. Then I can subtract. But look at what I'm going to do here. Did anybody change to a mixed number before doing this problem? Did anybody change to a mixed number?
I did.
Okay, that's another approach, which I know most people do that. But for this test, you don't want to waste too much time, because some questions will be easy to use that approach. But for some questions where you have multiple steps, let's say 2 or 3 steps, it may waste your time, doing that. Right? So look at what I'm going to do here. That may be more helpful as we go on. 5 minus 8 will be a negative number, right? But I can borrow. I have a whole number here that I can borrow from 6. So I can borrow from the whole number to make this numerator bigger before I can subtract. So if I borrow 1 from this 6, I'll have 5 whole numbers left. The one that I borrowed is always in terms of the denominator, right? So if I borrow 1, the one that I borrowed in terms of the denominator is 12 out of 12. That's the one that I borrowed. Everybody understands that? 12 over 12 is the same as 1. So whatever you borrow is in terms of the denominator, right? So I add 12 parts to the 5. So now I have 17 out of 12. That will be my new fraction. Is everybody okay with that part so far?
Yes.
The borrowing, yeah, because that will help you a lot if you have multiple steps. Because sometimes you have to subtract and add and can waste your time. Don't do. We change to mixed number. Okay, so minus now 2/8 over 12. So I've created a bigger fraction. Now I can subtract. So 5 minus 2 is 3, and then 17 minus 8, and then that will give me 9 over 12. Reduce to lowest terms. I can reduce 12 and the 9. 3 into 12, so 3 into 9 is 3. 3 into 12 will be 4. We have 3 over 4. So it's 3/4. Okay. So the borrowing will help you a lot if you can do that. Is everybody okay with the borrowing portion? At least for today, if you can remember that for fractions, subtracting fractions, that would be great. Everybody okay with borrowing for a whole number to create a fraction?
Yes.
Yes.
Okay, that's good. All right. All right, so that is how I will approach this question. All right, so let's look at number 5 to 10. Let's try that in a few minutes and then we'll come back to discuss 5 to 10. Again, number 5, you can use proportion. You're going to realize a lot of proportion can be used for so many things. So the American Diabetes Association estimates that 1 in 20 people will be diabetic at some point in their time. In a city of 120 people, how many are expected to be diabetic? So set up your proportion for this, and then I'll write it down. Okay, so for the first one, if you are diabetic, so if you have it this way, then you're correct, on the right track. So you have 1 diabetic out of 20 people. So 1 to 20. So the other ratio should be the same thing, diabetic to people. If you're consistent this way, you never go wrong. Okay. So here we have people, so the people go here. And then we don't know the number of people who have diabetes. So we can put this, our X here. So 1 over 20 is equal to X over 120. And then you can solve. It's 120,000. I left 1 0. Okay. And then you can solve for X. So you have 20 times the X is equal to 120 times 1, 120,000 times 1, cross product. Then divide by 20, divide by 20. So 20 cross out. So you have X is equal to, and then you have 6,000. So you should come up with 6,000 for this. So that would be B. Everybody had that?
Yes.
Would be, okay, good. So proportion. Okay. So what do you get for number 6? Number 6 should be straightforward.
C.
C. Okay, Cora, what did you do for that one?
I divided 3 over 20 and I got 0.15.
Oh, you work backwards.
Yeah.
Okay, so that's a good approach too to doing problems. You can work backwards to see if you can get what is given. So you choose the answer, take the answer and divide and see if you can get that. Good. Or what you can also do, this one is 15 out of how many parts? 1, 2, 2 decimal places, right? So if you do 2 decimal places, this is it's out of 100. Okay, 10th column, and that's 100th column. So it's 15 out of 100. That's what this means. It's like 15%, 15 out of 2 zeros. So that's 100. And then if you reduce to the lowest terms, 5 into itself, 5 into 15, that'll be 3, 5 into 20, and then into 100, sorry, it's 20. So it's 3 over 20. Okay. Or you can work backwards from the answer. Okay, okay. All right, number 7.
D.
D. Okay, she says D. What approach did you use for number 7?
I did it backwards also.
Great. I will do it backwards for this question. Backwards approach.
I got X. First, I got answer A, which is X equal 10 and Y equal 4.
Mm-hmm.
And it wasn't correct.
Okay.
And I did it step by step and I got to number D and it was correct.
Right. So backward approach for this question will be useful because there are 3 common ways of solving what we call the system of equations: elimination, substitution, and then you have the graphical approach. Those are the 3 common ways. The other ways, these are the 3 common ones. But since the answer is given to you already, as you said, you can work backwards to get the answer. So this is what I'll do. I create a table like this, and then you have to substitute the numbers into each of these. You should satisfy the first one and satisfy the second equation as well. Both should be true. If one of them is true, that is not true, then it's not correct. So if you take the first one, X is 10, Y is 4. So if you go 4 times 10 plus 3 times 4, sorry, 3 times 4, is this equal to 14? Let's see. This is 40. This is 12. It's a big number, so it's not equal to 14. This is not equal. So it means that this is not correct. All right, so it's wrong. Then try the next one, B, 8 and 26. It's going to be too big. You know, if I'm doing it, I don't even waste my time doing that at all. But it's going to be a big number. So why should I, waste the time? We can plug it in. But just for the sake of it, let's see. 4 times 8 plus 3 times 26. Too much of a big number. So it's again not equal. So it's not good. So it's going to be between C and D. So let's say C. What happens with C? 5 and 2. 4 times 5 plus 3 times 2, is that equal to 14? This is 10. This is 6. So this also doesn't work out. This is not equal to 14. So it doesn't work out. So it means that the answer is D, as she said. Let's test that quickly and see. So it's not good, not good. 4 times 5 plus 3 times -2 should be equal to 14. 20 minus 6 is equal to 14. This is correct, so it's good. You should also satisfy this one. Both should be true. There are some questions, this will be true and this is false. Then that cannot be an answer. It should be true for both. 6 times 5 minus 5 times -2, is that equal to 40? This is 30 plus 10, 40. So it is also correct. So it's true of both. So that's why answer D is correct. Okay. So backwards approach for this is helpful. Okay. Good. Yeah. Backwards. All right. So what about the next one? Number 8.
I got A.
You got A. Okay. Yes. So it's A. Milligrams to 1,000 milligrams is 1 gram, or 1 milligram is 0.0001 gram, whichever way you look at it. Okay. So it means that milligram is a bigger unit. So it means you have to divide. You expect a smaller number. So you have to divide by 1,000. Okay. But if you can't remember that, you can use proportion. As I said, proportion will work out for so many things. So let's say I can't remember that I have to divide by 1,000. I can still use my proportion. So I'll go and write, I'm given that 1 milligram is equivalent to 0.001 gram. That is given. This is equal to that. So I can set up the same proportion here, the same ratio. I need the milligrams here and then grams in the denominator. I've been given 256 milligrams. I'm looking for the grams X. Okay. So that's a proportion. If you're stuck, think about proportion. Okay. So that tells me that I need to multiply this by that. So 1 times X will be X is equal to 0.001 times 256. And then you get your 0.256 grams. Okay, so that's 8. Correct. Okay, so proportion will always help you out. Okay, good. All right, number 9.
I got B.
You got B.
B.
Okay, B. All right. So what approach did you use for number 9? 1/2 of X. So you can write that as a 1/2 X. Or translate to what?
I got D.
You got D.
Yeah.
Okay.
So
So
the
let's
easy,
read together and see what we get here.
alright, so the easy way I did it is basically, if you know 1/2 equals 64, right, it's looking for 1/8, right? So I did 64 divided by
No,
4.
it's
So
half
1/2 of
half of
X,
X equals 64.
64.
So it's
So what
32.
is X?
Yeah, half
So
of
X is
no, the thing
32.
No, look
I
at it
know
carefully.
if you divide 32 divided by in half again, you get 16.
Okay, look at this
So
again. This is the translation. 1/2 of a number, 1/2 X,
So
is
it's
equal to 64.
32.
What is X?
32.
Look at it again. Half of a certain number is 64. What's the
64.
number?
62, 64 times 2?
Yeah, 64 times 2. So you have to multiply both sides by 2 times 2. So we have 2 times 64. So the number you're looking at is 128. That is the X, right? Then
Oh.
now you want 1/8 of X. So it means 1/8 times the 128. That's what you're looking for. You got it? 1/8 of the X. So 1/8 times 128. So this gives us 1/6. So that's why it's 16. 16. So be careful with this part here. 1/2 of a certain number is 64. What's the number? The number is 128. It means 2 times this number to get X. We multiply both sides by 2. Okay, so answer is B. What about number 10?
I also got A.
A.
Same here.
Okay. So if you look at number 10, again, you can use proportion. So I'm going to mention proportion several times till you get tired of proportion. So we can have, it's a, you have penicillin solution contains 300 units. So we can do 300,000 units. So you have the units in 1 ml of solution. So it's 300 to 1 ml. I can set up the same ratio here. This will be units and then the ml. So the units here, we have 60,000. This is missing. I don't know the ml. So that's the proportion. So if I do the cross product, your X will be equal to, so that's 300,000. It's equal to 1 times 60, 60,000, divide by 300,000. Do the same thing on the left side, on the right side. So this will cross out. So you have your X is equal to, and then the zeros will cross out. So 4 zeros, all these 4 zeros out, we cross out 4 zeros here. So basically, you're left with 6 over 30. And if you reduce to the lowest terms, 6 into itself is 1. 6 into that is 5. So that's why you have 1 over 5. That's ml. So proportion, very, very helpful. 1 over 5. Okay. Okay, good. So look at number 11. Number 11 should be a quick one. Number 11. Square root of 64 over 4.
Is D.
D. Okay, good. So square root of 64 is going to be 8. So which number multiplied by itself give you 64? So that is 8. And then you do that first and then divide by the 4, and then you have 2. So that is D. That's correct. D is correct. Okay, good. All right. All right, take a minute to look at number 12. Look at number 12. So what do you think we should do for number 12?
There can be 2 ways you can do it. You can do 250 times 18%, that gives you 45. You can add 250 plus 45, or you can do 250 plus.
Okay, let me write what you're saying.
Yeah.
So first, you're going to find 18% of 250 that we're saying.
Yeah.
Right? So 250, 18%, we can change the decimal, so 0.18.
Yeah.
And then you had
45.
45. Okay, so that's the increase amount. And then, so once step 1 and step 2,
you add it.
You add on, correct. So you can add on 250, the price plus the increase. So think of when you're shopping, you know, percentage increase. So this will give you 290.
Or you can do 250 times 100.
This was hold on. It's 295, yeah.
295.
So that's $295. Okay. And then what else are you saying?
You can also do 250 times 118%, And
Yes,
it'll be factored away.
correct. So that one just one step is that you've added a percentage already. But I know most people will do it this way. What she's saying is that another method, if you are very confident, then you can use that method. 18% increase. So it's like, think of proportion. If I have, let's say, 100%, this is a percent proportion, right? 100% represents the whole 250. If you go 18 above 100, 18%, that means increase. You've gone 18 above 100. So the total percentage now will be 118. That's a new percentage you're talking about, 118%. Now, what would that be equivalent to? The new amount, you don't know. So it's X out of 250. X out of 250, you know. So if you use that method, the answer will just come out as this. So it's 118 times 250, and then we can divide by the 100, and then your answer will be 295. Yeah. Yeah. But I know most people will do it in the first method. Okay, great. Okay. So this is, at least for today, we've spoken about the idea of proportion to work out problems. We're going to use that a lot. So focus on proportion for today. And when we come next week, we'll use that to finish up the first math test. Next week, we'll spend more time on the math too. The science is a lot, so I use a lot of time on the science, but the math will definitely finish up. Okay. So just to round up for today, percent proportion, I'll leave you with the percent proportion that we can use for percentages as well. Remember, we have part over whole, which sometimes we call the base, is equal to the percent, and the base is always 100%, the percent of 100. So this is a percent proportion that I want you to think about. We'll use that a lot when we have problems with percentages. Part over whole is equal to the percent out of 100. And that's what I try to use in the second method. If I know the percentage, that percent over 100, and then the part and whole. You can, some people use the is and of. I don't know, most high schools, they will use the word is and of to help the students understand this. The part is always associated with the word is, and the base is always associated with the word of, so that can also help some people. For example, if I say John is a student of Hudson. If I say that, then I'm looking at a student of Hudson County Community College. So John is, that's a part. It's a part of, and the of is like the base that you're working with. So in every question, you want to identify the portion that is and the portion that is of, right? So something like, what is, let's say, 10%. Okay, let's make it this way. Let's say 15 is what percent of, let's say 100. Like this. So you identified the base and the part. 15 is, because the 15 is, that was a portion. So to 15 over of the base. So that's of 100. So that's 100. Then it's equal to, you're looking for a percent. So that's X, we don't know, out of the 100. And then you can work out the X, right? So this is the idea of using this formula to create a proportion for solving. Okay. So we'll pause here for today. A lot of information today. All right, so I'm going to post this recording on the Google Classroom. If there's something that you missed, you can always go back and look at it. So any questions before we stop for today? Any person, any question?
No, Professor.
No, right. Okay, so if you have a question, you can please email me. I will definitely respond to it. Thank you all. Nice meeting you all for first day of class. It's been great.
Thank you.
Hopefully next week we'll
Thank
take a break in between. You know, so it's a long session. We'll take a break in between so that we relax like 5 minutes in between next week. All right. Have a good day. Bye-bye.
You too, Professor. Stay
Bye-bye.
safe. Bye.
You too. Thank
Stay safe.
you.
Bye.